Dr. Mille.
Yes.
So we are now 1 o'clock. So let me start the meeting and introduce you. So welcome everyone to this first International Pediatric brain tumor course, neuro-oncology course. And this is a 4-day course. And we have got eminent speakers on these days. The first day we are going to have lectures on neuropathology and molecular biology. I think increasingly we all recognize this is going to be very important for us to understand to deliver the best care for the patient that is tailored to the patient. And on the second day, we will have lectures on all the main pediatric brain tumors from a faculty, again drawn from around the world. And on the day 3, we will have keynote lectures from neuro-oncologists and illustrious neurosurgeons. And 4th day, we will have discussions on controversies. So it's a great pleasure and privilege to welcome to you all to this course. And the first speaker who has put together this program is Dr. Douglas Mille. Dr. Douglas Mille is an eminent neuropathologist. He's a professor and chairman of pathology at the University of Missouri in Columbia. He was previously neuropathologist at the New York University at the time that Dr. Fred Epstein was making pioneering advances in the management of pediatric brain tumors. And before that, Dr. Mille had done his residency in MGH and his undergraduate studies in Miami, where his studies included philosophy. Dr. Mille, as you will very soon see, has an encyclopedic knowledge, insight into neuropathology. This comes from a vast experience of both research and both clinical work and also scholarship. His book on neuropathology for me is one of the best books that I have read. It is a single author textbook on surgical neuropathology, which is very insightful, and I hope he will bring out a new edition soon. So without further ado, I would like to invite Dr. Mille to give this lecture and then subsequently chair it, and then I will chair whenever Dr. Mille wants. Thank you very much, Dr. Mille.
Thank you, Naren, and good morning or good afternoon or good evening, depending where you are around the world, to all of you. So you can see my title slide, and Naren asked me to help organize this first day. He was very helpful in getting some of the other speakers lined up. So let me get started right away. So my talk for this morning will be about the WHO classification of pediatric central nervous system tumors. And my goals and objectives here is to lay a foundation for you, as you can read here. The WHO classification over the years since about 2000 has given neurosurgeons, neuro-oncologists, neuropathologists, radiation oncologists, and others a common language. We may not all agree on the classification's finer points or even how each case might fit, but we can use this common language to describe and classify tumors so that we can have common views on a large series on therapy and so forth. So my hope today is to lay a foundation for the rest of the course by giving you an overview of the classification. Some of you perhaps heard me talk on the subject of the new 2021 WHO version of this classification a couple of months ago. This talk is somewhat similar, but will concentrate on the pediatric tumors. I do want to note that all the photomicrographs that I will show are either copyright by me from my book that Naren has kindly mentioned, published in 2009, or from the article in Neuro-Oncology by David Lewis and colleagues describing the new 2021 version of the WHO classification. So to start, if we look at the gliomas, we have in the WHO classification adult type diffuse gliomas and pediatric type diffuse gliomas. And this is a new thing for the WHO. Rather than say that there are pediatric cases of something like oligodendroglioma or glioblastoma, they now have really separated these and said there are adult type and pediatric type. It's not quite the same as saying these are pediatric tumors, because they recognize that occasional children will have an adult type tumor, and young adults and occasionally even older adults will have a pediatric type tumor. And I'll just note for you that we recently had a case here of a 47-year-old man with a tumor that I called giant cell glioblastoma, but which by methylation array classification, and you'll hear from Dr. Aldape later this morning about how that works. By that, it was a pediatric type high-grade glioma with a receptor tyrosine kinase 1 subtype. So these can happen in adults, just as adult type tumors can happen in children. So among the pediatric diffuse low-grade gliomas, and Dr. Hawkins will talk about all the pediatric gliomas a little bit later this morning, we have these categories: diffuse astrocytomas, which have genetic alterations in either the MYB or less commonly the MYBL1 genes, something called angiocentric glioma, a relatively newly recognized entity called polymorphous low-grade neuroepithelial tumor of the young, which is usually referred to as plenty because it's easier to say. And this tumor can look a lot like oligodendroglioma histologically, and accounts for a lot of the cases that used to be called pediatric oligodendrogliomas. Diffuse low-grade glioma with MAPK pathway alterations, and this encompasses a number of tumors that could have been called astrocytoma or oligodendroglioma, depending on where you looked in the tumor or the particular case. And this would seem to end discussion about pediatric oligodendrogliomas, which are not IDH mutant and not 1p19q co-deleted, as the adult definition of oligodendrogliomas has it. And then there are the pediatric diffuse high-grade gliomas. One of the most important of these is the diffuse midline glioma with the H3K27 alterations. This used to be referred to as H3K27M mutant, but now the WHO is preferring to call this altered. There's a diffuse hemispheric glioma with H3G34 mutations. There's a diffuse pediatric type high-grade glioma with H3 wild type and IDH wild type. So this is not yet molecularly defined except by negative attributes. And then there's an infant type hemispheric glioma, and these have been reported with fusion genes that involve, in different cases, these other genes, especially the NTRK genes. So that's the landscape of pediatric gliomas. To show you some of this, among the low-grade gliomas, there's angiocentric glioma. These are usually cortically based. They present often, or most often, with epilepsy, with seizures. And histologically, they have this infiltrative appearance where there's a perivascular clustering of the tumor cells, often with what looks like almost pseudo-rosette formation, such that these things were originally recognized as possibly being cortically based ependymomas. In the 2016 WHO revision, they had said that all of the reported cases that had genetic analysis had a fusion gene with rearrangement of the MYB gene, which is on chromosome 6q, the long arm of chromosome 6. But subsequently, other tumors have been identified with some MYB alterations, as I've already stated. And so this is no longer regarded in the 2021 classification as a specific requirement for the diagnosis of angiocentric glioma. I'm showing you here a GFAP stain where there's these GFAP positive glial cells clustered around blood vessels in the cortex, as well as occasionally diffusely infiltrated through it. So that's angiocentric glioma, tumor with usually a pretty good prognosis. This is the plenty, the polymorphous low-grade neuroepithelial tumor of the young. The polymorphous part of the name implies that there can be different patterns in the same tumor. You can have areas that look like oligodendroglioma, as here on the far left. You can have areas that look like they're quite pleomorphic and astrocytic, which would suggest a high-grade appearance just by the nuclear variation in size, shape, and staining, as you see in the center panel. But these are usually low-grade tumors in terms of their clinical behavior and in terms of their proliferation rate. One helpful clue for neuropathologists and surgical pathologists is that these tumors exhibit a very strong and diffuse immunoreactivity for CD34, a marker that otherwise is useful for things like endothelial cells. So that's one helpful hint for that. We've known for a very long time that children in particular, and especially younger children, are susceptible to tumors that affect or seem to be centered in the pons. And these have been known as, quote, brainstem gliomas, which is not a very good name because there are other types of brainstem glioma. They've been known as diffuse intrinsic pontine glioma, or DIPG, and that name is still widely used. But now the WHO is defining these as midline tumors that are not just in the pons. They go up into the thalamus, they go down as far as the spinal cord. And these are being found to be almost universally of the H3K27M altered variety. And so that mutation in a midline tumor implies a very, very aggressive tumor with a very poor prognosis. I'm showing you some autopsy examples. This one goes all the way back to my residency, and you can see a vastly enlarged pond with hemorrhagic and necrotic areas where tumors replace the ponds, and there are metastatic nodules down in the subarachnoid space around the cord. That's a very common phenomenon in patients with this tumor. Another example here, a sagittal view. You can see the hemisphere and virtually the entire brainstem and much of the cerebellum replaced by this partially necrotic massive tumor. And then down here on the lower right, several cross sections of brainstem from midbrain down to medulla. The medulla appears mostly uninvolved. These can occur, as I said, other places. We've recently had an example in a young adult in the spinal cord. I need to caution you that H3K27M alterations are occasionally reported in other kinds of tumors that are not midline, including gangliogliomas, and in other situations it seems, also in ependymomas I should add, and in other situations it seems not to carry the same deadly prognosis that it has with these midline glial tumors. So the mutation has to go along with the anatomical and pathological findings. The next major group after the diffuse gliomas that the WHO now deals with are circumscribed gliomas. These used to be called the special types, especially the pilocytic astrocytomas, but now these are called circumscribed gliomas to imply that while they may be infiltrative, they're infiltrative only over short distances and to a minimal degree. So the most common and most recognized of these is pilocytic astrocytoma. Some of you will know that there has been an entity for years called pylomyxoid astrocytoma. This was almost always in the hypothalamic and third ventricular region and was seen mostly in very young children, in infants, and thought to be more aggressive than pilocytic astrocytomas, which is why it was made a separate entity or type. But it's turned out, number one, that genetically these are identical with pilocytic astrocytomas. And number two, that in children who have had resections and other treatment of these, who then get recurrences, the recurrences most often appear to have, quote unquote, matured into ordinary pilocytic astrocytomas. And so now the WHO quite rightly is regarding this as no longer a distinct type. They don't use the word entity, they like to use the word type in 2021. So pylomyxoid astrocytomas are just a variant appearance in pilocytic astrocytoma. There's an entity which is rather difficult to diagnose pathologically. It's called high-grade astrocytoma with pyloid features. These used to be called anaplastic pilocytic astrocytomas, or some variant on that, such as pilocytic astrocytoma with anaplastic change. These are more aggressive tumors. There's no distinctive molecular diagnosis in terms of specific genes or fusion genes or anything like that. They don't have the typical BRAF mutations that we see in many of the pilocytic astrocytomas. But this tumor type was identified using methylation array classifier. And so again, Dr. Aldape will talk about that method and how it's applied. But to make this diagnosis, a neuropathologist could suggest it from other features, but to confirm it really needs to have the methylation array classifier. Pleomorphic xanthoastrocytoma is a tumor that's been known for a great many years, first identified by the great John Kepes, who was a Hungarian who came to the United States in the 1950s and was for, I think, over 30 years a neuropathologist at the University of Kansas in Kansas City. This is still classified among circumscribed gliomas, even though it's widely known that these tumors, usually referred to by the abbreviation PXA, these are really glioneuronal tumors that they do express neuronal antigens in considerable variety. Nevertheless, these are slowly growing tumors, most often found in temporal lobe locations, and usually are pretty indolent. There are anaplastic variants of these that are more aggressive. And there's an overlap in pathologic appearance with what's called epithelioid glioblastoma. Many of these tumors have BRAF fusion genes. Subependymal giant cell astrocytoma, mostly associated with tuberous sclerosis complex, but not always. Another low-grade tumor. Chordoid glioma, a more recently identified entity, maybe about 20 years old, so to speak. Used to be thought to be mostly a third ventricular tumor, but now known not to be limited to that location. And finally, astroblastoma, an entity that has come and gone multiple times. It used to be controversial, but now it has a specific genetic alteration in MN1, and so the definitive identification of this, both in that it exists as a type and that it can be diagnosed specifically, is now advanced by our molecular understanding of these tumors. So to show you some of this, these 4 panels are to illustrate the major features of the usual pilocytic astrocytoma. Remember that these are most commonly found in the cerebellum, but they then are second most common site is in the hypothalamic suprasellar area, third ventricular area. They can be found almost anywhere, and even very rarely in intramedullary spinal cord locations. The classical pilocytic astrocytoma has what I grew up calling a spongy and compact architecture, where you have solid in an H&E stain, pink staining fibrillary areas interspersed in almost a checkerboard pattern with looser areas with lots of microcystic spaces. And the microcysts sometimes contain a fluid, which is eosinophilic, as you can see in the upper right. Some of these tumors will have eosinophilic granular bodies, these round or irregular granular eosinophilic staining structures, which appear to be extracellular, but I think by electron microscopy they're intraastrocytic. And then more commonly, they will have Rosenthal fibers. I put a couple of arrows to point to a few, but there are many pink staining Rosenthal fibers here. Rosenthal fibers are eosinophilic aggregates of filaments that are outlined if you do a GFAP stain, but they don't stain with GFAP. And again, they're intraastrocytic in this. They are not specific to pilocytic astrocytoma. They can occur in gliosis around certain other lesions. So characteristically around craniopharyngiomas in the hypothalamus, the glial tissue will have Rosenthal fibers. So you have to be a little careful about using those to identify something as pilocytic. This is to illustrate some features of pleomorphic xanthoastrocytoma. As the name implies, it's often quite pleomorphic, so your pathology colleagues can be, I'll use the word advisedly, frightened a bit by saying this looks very aggressive, and yet mitotic figures are scarce. The Ki67 labeling index will be low. So these tumors have a large number of large pleomorphic cells, sometimes binucleate or multinucleate, with generous amounts of eosinophilic cytoplasm. Interestingly, there can be deposits of reticulin or other kinds of collagen markers among some of these cells, and CD34 can be helpful for that also. In general, the cells are GFAP immunopositive to a considerable degree, but some of them also have neuronal characteristics, including immunopositivity for synaptophysin and neurofilament protein. The name xanthoastrocytoma comes from the fact that some of these cells are heavily lipidized. That is, they contain lipid vacuoles in the cytoplasm. And indeed, when Keppers first identified these tumors, he thought they were sarcomas rather than glial tumors, and he had called them xanthosarcoma. But then GFAP immunostaining technology came along, and when he found that they were all GFAP positive, he redid the name. Got more than one publication out of it. So I'm showing you here in the middle on the lower panel a cell which has a typical pleomorphic nucleus, and then around it the cytoplasm is vacuolated with these lipid droplets interspersed with glial filaments that are GFAP positive. This is a typical field from an ordinary subependymal giant cell astrocytoma. These cells are very characteristic. They're not pleomorphic, they're quite uniform, although the nuclei are large. The nuclei resemble a ganglion cell or large neuron nuclei. They often have a pale chromatin pattern with a very distinct and fairly large nucleolus. But the cytoplasm looks more astrocytic. It's quite eosinophilic. You don't see Nissl granule-like accumulations in the cytoplasm of these. These cells also individually express both GFAP and some neuronal markers in almost all circumstances. These are very low-grade tumors. In patients with tuberous sclerosis, where they are most commonly seen, the problem is that they occur at the foramen Monroe, cause obstructive hydrocephalus. And if you remove one, it doesn't mean that the patient won't get what seems to be a recurrence, but is probably a new primary, since they have an underlying germline genetic disorder in either TSC1 or TSC2. Chordoid glioma gets its name from a more than vague resemblance to chordoma, although this is clearly a glioma. The background looks almost like cartilage with a mucinous or myxoid ground substance in the extracellular spaces, and the cells line up in these linear cords as in chordoma. The cells are usually not vacuolated, unlike the cells of chordoma, so there's really usually no confusion about what these are as compared to chordoma. But the name was given because of this histologic resemblance to chordoma. And astroblastoma. So astroblastomas are often cerebral hemispheric tumors. There are sometimes other gliomas which have a pattern that looks focally astroblastic, and that has been part of the confusion over the years. But pure astroblastomas have this diffuse pattern, although they end up being circumscribed at the edges, with this characteristic perivascular pattern that looks like the pseudo-rosettes of ependymomas. But if you look at high magnification, the processes don't taper, they're broad all the way down to the vessel wall. And that is the characteristic that has led to a differentiation histologically of astroblastoma from ependymoma. Subsequently, as I have already said, there's a distinctive genetic alteration in the MN1 genes, which allows one to verify that a particular tumor fits the WHO criteria for being an astroblastoma. Okay. Next, we turn to glioneuronal tumors, or just plain pure neuronal tumors. And this group of entities keeps growing. So it used to be we had about 3 of them. We had ganglioglioma, gangliocytoma, and neuroblastoma. And now we have all these others that have accumulated over the years. So some of these favorite entities are still with us: ganglioglioma, desmoplastic infantile ganglioglioma, or astrocytoma. These are really the same entity. Dysembryoplastic neuroepithelial tumor, as first described by Catherine Damantepour in Paris some years ago. A new entity, diffuse glioneuronal tumor with oligodendroglioma-like features and nuclear clusters. This is in italics, as I've written in white font, because in the WHO classification in 2021, they have a number of entities they call provisional types. And so they haven't fully accepted that these are real. They're still looking for some more verification, but they felt there was enough to put them in the classification. So this is a provisional entity. Very rare tumors. I think there are about 19 or 20 in the literature right now. Older entities that are well recognized is papillary glioneuronal tumor, the rosette-forming glioneuronal tumor, something called myxoid glioneuronal tumor, a tumor type that was identified about 15 or 20 years ago, although obviously some of us saw cases before then we didn't know how to classify, called diffuse leptomeningeal glioneuronal tumor. I'll get to show this a little bit more sometime, but basically these tumors look a lot like oligodendroglioma with round nuclei centrally placed in clear cytoplasm, and they grow either in multiple foci or diffusely in the leptomeninges. So you think the patient has a diffuse metastatic disease, but it's actually a primary leptomeningeal glioneuronal tumor, which can be multifocal. There's gangliocytoma. There's something new called multinodular and vacuolating neuronal tumor that's only about half a dozen years old. The famous dysplastic cerebellar gangliocytoma, or Lermite-Duclos disease, which has been known for almost a century, I think. Central neurocytoma, first described by Jacques Hassoun in about 1982. The extraventricular neurocytomas, which are still hard to define in either a pathological or molecular way. And finally, cerebellar liponeurocytoma. And I'm a little puzzled by the WHO committee's decision to call it that, since liponeurocytomas have rarely been described in non-cerebellar sites. But they've retained cerebellar in the nosology in the 2021 classification. So this is just one H&E field from a typical ganglioglioma. You see in a background a majority of small astrocyte-like glial cells, maybe some microglia with these elongated nuclei, and then interspersed among them these larger cells, which have sometimes multinucleation, sometimes single nuclei. The nuclei often are clearly neuronal with a large nucleolus, and the cytoplasm is sort of amphiphilic. It has some blue staining as well as some pink staining, and these will stain with neuronal markers. Particularly if you use a synaptophysin immunostain, you'll get decoration of the outside of these large ganglion cells, a pattern that I first described back in 1991 when synaptophysin antibodies were relatively new. These are indolent, slow-growing tumors. They often present with epilepsy, but you can find them anywhere in the nervous system. The literature concentrates perhaps on the temporal lobe examples, but they certainly can occur in the spinal cord. They can occur in the cerebellum. They can occur in other lobes of the hemispheres. Papillary glioneuronal tumors are relatively rare but well-described entity. It's a rather peculiar tumor in some ways. You get this papillary, or what some would prefer, pseudopapillary pattern of cells clustered around thick-walled vessels, and then with a more diffuse pattern in between those vessels. The background in between vessels is synaptophysin immunopositive in the cytoplasm and in the processes of these cells, demonstrating that they are fundamentally neuronal. But around the blood vessels, the cells that form a tight collar on the blood vessels, there's GFAP immunopositivity. So something about the relationship to the vessels causes a different differentiation in tumor cells that share common origins with the neuronal elements that are more diffuse. These are, again, low-grade and usually resectable. DNT, described, as I said earlier, by Catherine Damas-Duport, first in the French literature and then in a what's been regarded as the landmark paper in the English literature done with Bernd Scheitauer at the Mayo Clinic. She spent a year at Mayo and went through their files and found that they had a bunch of these that they hadn't classified correctly because it was a new classification back then. So these are typically multicystic tumors that show up with a very bubbly pattern on MRI. Histologically, you can see these microcysts with a relatively dense pattern of small cells in between, usually without obvious ganglion cells except inside the cysts. Inside the cyst, you can have what are called floating neurons, where there are large neurons that just seem to be sitting there undisturbed by the fact that they look disconnected from the rest of the environment. The cells in between have round nuclei, often some clear cytoplasm. They look like what we call oligodendroglioma in other circumstances. And indeed, Dr. Damas-Duport originally described these as oligodendrocyte-like cells. We now know that these are really neurocytic cells that express neuronal markers and not glial markers. And there's some complex genetics that are known to be associated with these. The rosette-forming glioneuronal tumor is a very distinctive tumor, again, not very common. You can see a sort of a diffuse growth within the tumor, although these are, again, usually fairly circumscribed. But inside them, you'll find these neuropil-like clusters surrounded by tumor cells that project processes into them. And while that may not be entirely clear in an H&E stain, if you do something like a neurofilament or a synaptophysin, and I'm showing you synaptophysin, these rosette areas light up very strongly. So they're full of synaptic vesicles, synaptic endings, synaptic membranes. And sometimes you see these ganglion cells around them with processes projecting into the rosette-like areas, as I've shown on the right. Multinodular and vacuating neuronal tumor, the name is descriptive of what we see. So these holes are not histologic artifacts. I think the first impression a pathologist would have is that he or she got bad sections from their laboratory, but in fact, these holes are real. There's an MRI correlate with some T2 or flare intensity in these tumors, and in between cells it looks sort of astrocytic with eosinophilic cytoplasm and eccentrically placed nuclei. But in fact, these stain with neuronal markers, not glial markers, and so this is classified as a neuronal tumor. A relatively newly described entity is a myxoid glioneuronal tumor. These tumors have a very characteristic location. As we see more, I suppose we'll see some in other places, but they usually occur as an intraventricular mass in the frontal horns attached to the septum pellucidum. And this MRI appearance should make you think about central neurocytoma. So these tumors are a mimic of central neurocytoma clinically until we get to the pathology. Pathologically, they often look something like DNTs. They have microcysts. They have oligodendrocyte-like small neuronal or neurocytic cells scattered, making up the bulk of the tumor, apart from these myxoid material-filled microcysts. These tumors have become a distinctive entity because they have a distinctive genetic change in the platelet-derived growth factor receptor A. There's a dinucleotide mutation that characterizes this group of tumors, this type of tumor. So again, a molecular knowledge has now allowed a specific classification of something that previously would have been called something else. This is a typical example of the Wernicke-Duclos disease, a dysplastic gangliocytoma. You get large dysplastic ganglion cells, which are actually derived, we think, from mutant small granular neurons, not from Purkinje cells. And this disrupts the architecture of the cerebellar foliar cortex in the places where this tumor occurs. Okay, so having gone through those categories, we now get to the category of ependymomas. In the 2016 classification, the WHO said that there were 9 types of ependymoma, looking at the molecular genetic landscape, 3 each in the supratentorial compartments, in the posterior fossa compartment, and in the spinal cord compartment. We now have 10 types, and I've listed them here. So there's something which is just called supratentorial ependymoma. Then there are 2 types of supratentorial ependymoma that are distinct with distinct fusion positive alterations in either YAP1 or ZFTA. These used to be in the 2016 classification lumped together with a RELA fusion gene notation, but now we know that the fusion partner is different in these 2 different types. Again, posterior fossa ependymoma can be a category on its own, or there can be the PFA and PFA groups, PFA and PFB groups, which have somewhat different prognoses, and so it's important to make these distinctions. In the spinal cord, again, spinal ependymoma, there's a group of those which have an amplification of the MYC-N gene. There's some that have NF2 mutations. I didn't put that at the list here. And then, of course, there's myxopapillary ependymoma, which is almost always a spinal cord tumor, sometimes referred to as a cauda equina tumor, but it can occur at any level of the cord. And in extremely rare circumstances, it's primary in the intracerebral or intracranial compartment. And then subependymoma, which can be cerebral, posterior fossa, or spinal cord. So the pathologist can look at these and say, these are ependymomas, can distinctly identify subependymoma or myxopapillary ependymoma, but otherwise really needs to have the genetics to identify the specific type. And then, as I've said before in other occasions, we pathologists and others don't really know how to classify choroid plexus tumors except as a separate group by themselves, but they're always discussed with the ependymal tumors because they're intraventricular. And there are 3 types which are standardly recognized. Most common, the benign choroid plexus papilloma, rarely the choroid plexus carcinoma, which can occur in adults or children. And then there's a variant in between, which is atypical choroid plexus papilloma. And this is still a strange entity in my view, because if you do a complete resection, the patients are cured just as they are with choroid plexus papilloma. But if you do an incomplete resection, you get recurrences and sometimes metastatic spread. So ependymomas all look alike except for the myxopapillary and subependymoma types. In uncommon examples, you will find what are called true rosettes, or Flexner-Wintersteiner rosettes, if you like old eponyms. You get a flower-like, that's why it's called a rosette, arrangement where cells are arranged radially around a central structure. That structure is a lumen formed by the cells themselves, as if they were forming a gland or a canal, or, as I like to think of it when I teach, a miniature ventricle. These are ependymal cells that are lining a miniature ventricle. And so you see these examples. Not every ependymoma has these, but what ependymomas do have most commonly in almost all examples are perivascular pseudorosettes. I mentioned these when we talked about astroblastoma a few minutes ago. So these have these radial arrangements around a central structure, but the central structure is a blood vessel with endothelial cells. So that's different from the true rosettes. These are pseudorosettes. And the processes of the cells that taper, that go down and project onto the vessel wall, taper to very fine little points. So that's different from astroblastoma. A key factor here is that there's this zone of processes, which is not quite completely, but mostly nucleus free. So there are very rare nuclei in here. Mostly it's just pink processes. You have to be careful as a pathologist to not just be looking at a thick-walled vessel with a collar of collagen and cells arranged around the outside of that. So you have to prove these are glial processes. And that's easy to do with a GFAP or a vimented immunostain. A helpful thing in ependymomas is that because they're acting as an epithelium of sorts, they will usually have some immunopositivity for epithelial membrane antigen. And in difficult cases, there's often an intracytoplasmic sort of a perinuclear dot of EMA positivity, and that helps you identify what by electron microscopy is an ultramicroscopic intracellular lumen with microvilli and other characteristics of a typical glandular type lumen. This is a view of a myxopapillary ependymoma. To the uninitiated, including, let's say, pathology residents, this can be very difficult to diagnose because it doesn't look at all like an ependymoma at first glance. We have these myxoid lakes with thick-walled vessels and then just tumor cells in between. It's a distinctive pattern, and in some cases it does show some perivascular pseudorosette-like formations. For example, here there's a vessel in the center, the lower left of the picture, and you can see these cells arranged around it in a radial fashion. That's a little subtle, and you have to learn to recognize this pattern as a pathologist. And this is subependymoma, a very low-grade tumor under almost all circumstances. We like to characterize these for our residents as a sea of pink with islands of blue, meaning in H&E stains that there's a lot of pink fibrillary background with almost no cells, and then these loose clusters of nuclei, which are bland, small nuclei. It's not easy to see these as ependymomas at first glance, and there's an old classification called these glomerate astrocytomas. But if you analyze these, there's often in these seas of pink a vessel. The cells are sort of loosely arranged around them, what I like to think of as very loose, large pseudorosettes. This is just to show you a choroid plexus papilloma. It looks a lot like choroid plexus, but it's more complicated in its papillary folding patterns. You have epithelial cells arranged around the outside of the individual papillae, which have a loose fibrous matrix and a central blood vessel. Definition of a papillary structure with a main stalk that connects back to the rest of the choroid plexus. These can be quite bulky, and on occasion they can shed cells and have implants elsewhere. So they're technically potentially metastatic through CSF pathways, but they're all very indolent and not usually a clinical problem. And this is a choroid plexus carcinoma. So you can see it's still showing some epithelial behavior and growing around blood vessels in that fashion, but now it has extensive solid areas, which is not papillary. These tumors can have areas of necrosis, and they will invade the brain. You will see destructive invasion through the ependymal wall into brain tissue from a choroid plexus carcinoma. The next major category is embryonal tumors, and you can see there's a lot of those, but the most important category, both in terms of numbers and everything else, is the medulloblastomas. So if you know something about the history here, medulloblastoma was a word that was invented by Harvey Cushing and Percival Bailey. They had these primitive-looking tumors that they wanted to call something else. They wanted to call them spongioblastomas, but they found out that term was already used, so they invented the term medulloblastoma. There is no embryonal or developmental cell type known as a medulloblast, just because. We now, by definition, restrain this diagnosis or confine this diagnosis to posterior fossa cerebellar tumors. Histologically, over the years, we've developed three classifications: the classic ones, the desmoplastic nodular ones, a subtype of those with extensive nodularity, and then an entity defined originally at Hawkins, I think, as large cell anaplastic. Large cell was defined and anaplastic was defined, and then these two terms were combined. And so pathologically, a neuropathologist looking at these can make one of these diagnoses in a medulloblastoma. But genetically, we now are up to 9 subtypes. So in 2016, it was recognized that there were some that were activated by alterations in the wingless, using the Drosophila gene named the wingless or the Wnt pathway. There were others that were sonic hedgehog pathway altered, and there were those that had other kinds of genetic alterations which were more aggressive, which were called group 3 or group 4. And the genetic changes for these are still more complex. But now we know that among the hedgehog ones, there are those that are p53 wild type, and there are now 4 subtypes of those. And then there are the p53 mutant, which are known to be more aggressive. And even in the 2016 classification, it was noted that p53 mutant hedgehog-activated medulloblastomas were more aggressive than the hedgehog-activated ones that were p53 wild type. These tumors are almost never fatal. They have a really astonishing Kaplan-Meier curve that is right up at around 95%. It stays straight horizontal for many, many years. The Wnt-activated tumors are also relatively slow growing, and there are now active clinical trials, I think, in both Europe and America. I'm involved in the one here in North America as a review neuropathologist, giving reduced treatment for children with Wnt-activated medulloblastomas to see if we can preserve the good survival while improving the neuropsychological and endocrine consequences. There are other CNS embryonal tumors. Atypical teratoid rhabdoid tumor, defined first by Lucy Rourke and Jackie Beagle, who was a cytogeneticist when they were both at Children's Hospital of Philadelphia. We now have 3 molecular subtypes of ATRTs. Something new called cribiform neuroepithelial tumor. Again, it's in italics, meaning that WHO calls this a provisional entity. The embryonal tumor with multi-layered rosettes, as of 2016, this took over tumors that were previously called the etantar for embryonal tumor with abundant neuropil and true rosettes, and also the tumor type that was called medulloepithelioma. So these are now all lumped together as ETMRs. There are 2 of these. One has a recognized alteration in something called C19MC, and the others, which are less frequent, have Dicer mutations. There's really a third group which has neither, which does not have a defined genetic type yet. There's now something called CNS neuroblastoma FOXR2 activated. And so some of what we used to call PNET would now be this entity. There's a CNS tumor with B-core internal tandem duplications. These are quite rare. And there's a note in the WHO that they might end up calling these sarcomas instead of glial neuroepithelial tumors because there are B-core mutations in some other mesenchymal neoplasms. There is something called CNS embryonal tumor not otherwise specified, where it doesn't have defined genetic alterations, or not elsewhere classified, where the genetic alterations don't fit the established classification as of now. And I will note for you that the pineoblastoma is a embryonal type tumor, but the WHO doesn't classify it there. It classifies it with the pineal tumors. It's still an embryonal type tumor. So here's classic medulloblastoma, diffuse, sort of a monotonous group of small tumor cells with large nuclei for the amount of cytoplasm, negligible cytoplasm in most stains. The nuclei don't overlap each other. They don't wrap around each other. There are a lot of mitotic figures, but the mitotic rate is lower than in what we call anaplastic. They are often lined up in this sort of fashion. Older terminology, this used to be described as Indian filing, as an analogy to the mythology of the American West, where Indians would stalk through the woods, American Indians would stalk through the woods in single file along narrow pathways. This is a rather fanciful description, and we don't use it anymore, but I'm mentioning it because you can see this in this picture. The nodular desmoplastic medulloblastomas have these interesting islands that have abundant neuropil, larger cells with more neurocytic appearance of the nuclei. And these areas are very strongly synaptophysin or neurofilament immunopositive. The surrounding more embryonal areas are less so. And so this is the nodular desmoplastic type, which is almost exclusively associated with hedgehog pathway mutations. And then the anaplastic medulloblastoma, you see the nuclei are larger than classical medulloblastoma. They often wrap around each other in complex ways. The nuclei are more pleomorphic. These have higher mitotic rates. They tend to have more necrosis or apoptotic zones than the conventional or classic medulloblastomas. Atypical teratoid rhabdoid tumor, a rather pleomorphic or polymorphous tumor, often with very large epithelioid cells with big nuclei, prominent nucleoli, sort of a glial background in places. This can be mistaken for high-grade glioma in some cases, or for medulloblastoma in others, especially, of course, if it's in the posterior fossa, which is the most common site. These tumors are defined by a loss of function of the SMARCB1 or INI1 gene, and we use an immunohistochemical surrogate for that diagnosis. So this is an INI1 immunostain. Normal cells that is endothelial cells that are not neoplastic will have nuclear immunopositivity. The tumor cells lack it. There are uncommon examples of this, which have an intact SMARCB1 but have a mutation in another gene. We won't get into that now. It's in the WHO classification. But you have to be careful. If it looks histologically like this and it has not lost INI1, you want to make sure it doesn't have the loss of the other gene to get involved in making the right diagnosis. We're almost at the end here. This is embryonal tumor with multilayered rosettes, two different views. On the left, you see the abundant neuropil, and then these clusters around vessels and the rosette formation that led to the name of etantar that is now obsolete. And on the right, you see the medulloblastomas-like pattern with these epithelial clusters or lines of cells. Pineal tumors, I'm not going to show you these, but just briefly, there's pineocytoma, which is almost like a neurocytoma. It's very bland and often curable by surgery alone. There's the pineal parenchymal tumor of intermediate differentiation, which is between this and pineoblastoma, which is a embryonal tumor type. Now, the pineoblastomas have been subdivided by molecular changes. So you can see these. I'm not going to read them out for you, but they're complex. So you get an altered processing of some microRNAs together with either a dicer mutation or a drosha mutation or a DGCR8 mutation. And these are mostly in children. Some of the others are in older children, which different microRNA processing alteration. There's the FOXR2 MYC gene activated ones, which are mostly in infants. And there are those that are retinoblastoma altered. These are the ones we knew about most for the longest time. Infants with a germline loss of retinoblastoma will present with what's called trilacteral retinoblastoma, with retinoblastomas in each eye and in the pineal region. There's also something called papillary tumor of the pineal region, and then there's a relatively new entity called desmoplastic myxoid tumor of the pineal region, which has a SMARCB1 mutation like the ATRT. So that mutation is not unique to ATRTs. Notice that I haven't put the germ cell tumors here, and I'll talk about that briefly for just a second in a few minutes. The WHO classification certainly includes meningiomas, schwannomas, and neurofibromas, and these are tumors that mostly occur in adults. They can be found in children, and of course, if children have NF1, they will have neurofibromas. If they have NF2, they will have schwannomas and meningiomas, although they usually develop those later in life. But the fact is, I'm not going to illustrate these or talk about them more because they're mostly adult tumors. And this is, of course, on pediatric neuro-oncology. There's a group of mesenchymal neoplasms in the classification. These occur in adults and children. Sorry, I got to make sure that my messages are shut down. You can see and read these lists. Some of these we are well familiar with, like hemangioblastoma or other hemangiomas. I'm not sure some of the hemangiomas should be regarded as neoplasms, whereas hemangioblastoma clearly is. Other well-known entities, rhabdomyosarcoma, well known to occur rarely in the central nervous system. But some new things: intracranial mesenchymal tumor with a fusion gene involving FET and CREB. That's another provisional entity. The CIC rearranged sarcomas that are defined that can affect other sites, but also the central nervous system. The primary intracranial sarcomas with dicer mutants. And there's something called the dicer syndrome, where there's a germline dicer defect, which get multiple tumors which have dicer mutations in them. And then Ewing sarcoma rarely presents in the brain or spinal cord, but can. So we have to understand that entity also. So briefly, intracranial mesenchymal tumor with the FET-CREB fusion genes. This is spindle cells, a loose sort of myxoid background. CIC or rearranged sarcomas, rather cellular view here, high mitotic rate. And then the dicer-associated sarcoma with larger cells, often multinucleated. These can be quite pleomorphic, and you have to have the molecular information to make the right diagnosis. The WHO now includes in the central nervous system volume some bone and soft tissue tumors that involve the nervous system. So chondrogenic tumors, mesenchymal chondrosarcoma and chondrosarcoma. Mesenchymal chondrosarcoma is a weird entity because it can occur with no association with bone or cartilage inside the brain. And then there are notochordal tumors, of which there's really only one example, and that's chordoma, and we all know about chordomas. More often in adults, but can be seen occasionally in children. Lymphomas and histiocytic tumors. Most of the primary CNS lymphomas are in adults. We very rarely see any of these in children, but we do see histiocytic tumors in children, including the well-known syndromes of Langerhans cell histiocytosis, Erdheim-Chester disease, and Rosai-Dorfman disease. And obviously, juvenile xanthogranulomas occur more in children than adults. Histiocytic sarcomas in any age. Tumors of the cellular region, important things to know about. We're winding down on time here. We have 10 minutes. Sorry, I didn't need to go that fast. So the tumors of the cellular region that we should know about for children, craniopharyngiomas, two types defined histopathologically, but also with molecular differences, as the adamantinomatous variety and the papillary type. These are more in adults, the adamantinomatous variety about half in children, half in adults. Pituocytoma, granular cell tumor of the cell, and spindle cell oncocytoma, these all used to be regarded as separate entity, but now they are regarded as variants or subtypes within the within the same single tumor type called pituocytoma, or pituocytic tumors of the pituitary stalk and neurohypophysis. They still separately classify them. That is, they list them separately. Then there are pituitary adenomas. The WHO is trying to make the community, and this is mostly being pushed by endocrine pathology specialists, to change the name here to PITNET for pituitary neuroendocrine tumor. I don't know if that's going to catch on. I think those of us who've been calling things pituitary adenomas for 100 years are probably not going to change. And then there's a relatively newly defined or better recognized entity called pituitary blastoma. Again, germ cell tumors are not mentioned here by me, but they also occur in this site. So craniopharyngioma on the left, two views of a typical adamantinomatous craniopharyngioma. You have this epithelium, which is basaloid, very dense small cells, and then abrupt maturation, if that's the word, to these large plates of keratin, which are very pink staining, and these get desquamated into the lumens of cysts inside the tumor, producing what's sometimes referred to as a crankcase oil-like material that the neurosurgeons can suck out. Here's a higher magnification view of the epithelial components here. In between the keratin plates and the basaloid epithelium, we have a loose arrangement of cells which are in fact epithelial. This is called the stellate epithelium. These are keratin positive with immunostains. And squamous papillary craniopharyngioma on the right, you get these papillary fronds lined by a true squamous epithelium with a basal layer and maturation in the typical fashion of any squamous epithelium. These are benign on both sides, but because of their location, they are often a cause of significant morbidity and mortality in the long run. Pituocytoma on the left. I'm sorry that picture is blurrier than I thought it was, but it shows these short spindle nuclei in a fibrillary background. And then here from an autopsy of mine, a typical granular cell tumor, a very circumscribed small lesion in the pituitary stalk. To orient you, that's an optic tract. This is the anterior part of the third ventricle. This is going down anterior to the mammary bodies, and there's this tumor sitting right here. These are very strongly PAS positive, and this is a PAS Lutzel-Fast blue stain, just to illustrate that. This is pituitary blastoma on the left in H&E, a sort of a myxoid background, almost cartilaginous background with these epithelial rosette-like or gland-like clusters and sheets of cells forming trabecula. And the clue here for the neuropathologist is if you do an immunostain for adrenocorticotropic hormone or ACTH, you will find that some of these cells are ACTH positive. I've several times mentioned germ cell tumors. All the types of germ cell tumor that we know of in the gonads or in the mediastinum also occur in the intracranial compartment. Most usually either in the pineal region or in the suprasellar hypothalamic region, but occasionally in other sites, including basal ganglia. And so this is the full range of these tumors. I'm not going to read this out to you. You can see it faster than I can talk about it. I will mention that in the gonads, any of these tumors are highly curable with surgery, then radiation, or to a slightly lower degree, but still a high percentage of cases with chemotherapy. In the head, germinomas are highly curable as well. And so the usual practice that you will know well as neurosurgeons is biopsy these, verify the diagnosis. Often there are marker studies which help make the diagnosis before surgery, and then radiation or chemotherapy as the patients and families choose. But these tumors remain a survival problem, with survivals being somewhere in the 40% to 60% range despite best therapy. So these are more aggressive, despite the fact that in other body sites they are as curable as germinomas. And that sums up this talk. So I'm going to stop screen share.
Thank you. Thank you very much, Professor Dr. Douglas Mille, for once again this excellent lecture covering the neuropathology, the landscape that's relevant to us. It's an amazing lecture. It's, very useful reference. I just want to ask, there are a few senior panelists here, whether they have any comments or questions. Professor Goel, do you have any questions for Dr. Mille? Thanks.
No question, no question about the Professor Douglas Miller's work, his lifetime work. He has presented in a brief kind of summary what he has given a lesson in neuropathology, present, past, and future. And he has incorporated all the three facets of neuropathology into his lecture. And he has, given a summary as to what is happening in this subject and what is going on and what is the future. In general, I think this is a very, very interesting and important topic of discussion for all the young and senior neurosurgeons all over the world. I think this lecture should be kept in the archives of our, if you are keeping some kind of archives for the future reference for all neurosurgeons and neuropathologists all over the world. Congratulations, Dr. Mille. I know you, I have heard about you for a long time, and it was for the first time actually that I actually heard your entire lecture, Douglas. Thank you very much.
You're welcome, and thank you very much for the compliments. I've known of you from the Listserv for many years as well, and very well known and very well respected. There's one question in the chat about a temporal lobe leiomyoma. I would say that's vanishingly rare. Leiomyosarcomas are described. I think they're more often dural-based than intraparenchymal, but they are described. But a leiomyoma, a benign smooth muscle tumor in the brain, I suppose would arise from vascular smooth muscle cells. But I don't know of an example myself, and I don't think it's mentioned in the WHO. So for whoever asked that question, that's the best I can do for you here.
Thank you. I'll just ask Dr. Bandopadhayay, do you have any questions or comments from the neuro-oncology world to clarify? Thanks.
No, I am. Dr. Mille, the second half of your talk, of course, it was a wonderful talk, and really shows that I think the not challenges, but I think it shows the challenges, but also how much we've learned over the last 10 or 15 years. I'm always struck when I was training to be an oncologist, we didn't understand the biology at all. And within only 10 or 15 years, the community has learned so much, and I find that really inspiring.
Thank you.
I agree.
Thank you.
Now, if there's no more to do,
I think that I will just ask Dr. Alan Cohen. Dr. Cohen, do you have any questions or comments?
Thanks, Naren. No questions, but I just want to make a comment that, I knew Dr. Miller before he was famous. When I was doing my training, he started his career at NYU Bellevue. And this is the second time I've heard this presentation. And it's just a fantastic review of the new CNS5 system, which is revolutionary in that it's an integrated layered hybrid where there's both the old school of histology and morphology and the new school of the molecular biology, which has the opportunity to revolutionize the treatment by enabling targeted therapy. One of the things, though, Doug, is that with this layered diagnosis, often we have to start treatment before we have the final pathology. And this is not available worldwide. I think the world is becoming a smaller place, but any comment?
Sorry, Dr. Cohen, could you unmute again, please? Sorry, there's an interruption.
I don't know what, I'm just wondering, worldwide, this will have an impact, but not all places have the ability to do this. And I know some of the pathologists are allowing this to be done. They contact the relative pathologists. How do you think this is going to impact neurosurgery and neuropathology worldwide?
this is a real concern. So first, even in our own country, not every center can do this in-house. So we send them out to established laboratories elsewhere, and it takes time, and it does delay therapy to get to the precise diagnosis. I, in fact, am preparing with my colleagues here a proposal for us to install an in-house NGS facility precisely to shorten the turnaround time. It's not that we'll make money on it, we'll probably lose money on it, but we need to shorten turnaround time to benefit the patients. So this is true, and it's going across all oncology. It's not just neuro-oncology. And it's one of the pleasures of doing this, and thank you, Naren, for asking me to do it, is to see old friends such as Dr. Cohen, even if it's virtual. I think I should turn to introduce the next speaker.
Please. Thank you very much, Dr. Cohen. Thank you very much, Dr. Miller. Please, Dr. Miller, to introduce the next speaker. Thank you.
So our next speaker, I constructed this morning with Naren's collaboration to go from this overview of the molecular landscape of the pathology classification to some basic neurobiology, basic molecular biology. And then we'll talk a little bit about, Dr. Aldape will talk a little bit about methylation array classifiers, which is a relatively new thing. And then get to specific entities, Dr. Hawkins talking about gliomas, Dr. Fehlings and Dr. Samuel about ependymomas, and Naren will finish up talking about medulloblastomas and perhaps a word about some of the other embryonal tumors. So our next speaker is Dr. Pratiti, and I'm going to mess this up, so I'm sorry, Bandopadhayay. She is a pediatric neuro-oncologist at Boston Children's and the Dana-Farber Cancer Institute and Boston Children's Cancer and Blood Disorders Center. She's currently assistant professor of pediatrics at the Harvard Medical School, earned her MBBS at Monash University Medical School in Melbourne in Australia, did a pediatric residency and a fellowship in pediatric hematology oncology at the Royal Children's Hospital in Melbourne. She then came to the United States, did a fellowship in pediatric neuro-oncology at Dana-Farber and Boston Children's, and that finished up, I think, in 2013. She's an associate member of the Broad Institute of MIT and Harvard as well, and has an active laboratory in addition to being a working pediatric neuro-oncologist. And I will mute myself and turn it over to you.
Thanks, Dr. Miller, and thank you to everyone for the very kind invitation to present. I'm really excited to virtually be here. Let me just quickly just share my screen, and hopefully you can see my slides. Do they look all right?
You have to put it to the.
Hey, is it in presenter view or is it?
It's on presenter view at the moment. Yeah.
So it looks okay.
No, it's that's fine now. Yeah, that's great.
Yeah. So as Dr. Miller said, I'm a pediatric neuro-oncologist. I'm Australian, but I've been in Boston for just over 10 years now. And I have the privilege of leading a lab that applies genomic approaches to study childhood brain tumors. And what I thought I'll do today is just give an overview of how I think some of these genomic findings, the new classifications that Dr. Miller presented, is starting to impact the clinical care of children. And some of the challenges that we face, as Dr. Cohen said, worldwide actually, to how do you integrate this when you have a child sitting in front of you in the clinic, and some of the advances and some of the challenges. So before I go into that, this is just my disclosure slide. My lab receives grant funding from Novartis for unrelated work and has received funding from Deerfield and QED Therapeutics. So this is very well known to this group, but I always start talks like this with this because to me it is why we're all here. More children are diagnosed with brain tumors than any other solid tumor. And even though there's been tremendous advances in some of the other diseases, we have not seen the same translation of improved survival in children with brain tumors. So more children die now of brain tumors than any other cancer. And the other thing that I think is often overlooked when we talk about survivals is that those kids that survive are absolutely devastated very frequently, right? So we talk about 70 or 80% cure rates of medulloblastoma, but that's at a huge cost with high-dose craniospinal radiation, chemotherapy, and then or the subsequent sequelae, including second malignancies. And when I sort of was training, so this will age me, the year I graduated, actually, I graduated in 2001, the human sequence, the sequence of the human genome was published for the first time. And as you saw in Dr. Miller's talk, that has really changed our understanding of the last 2 decades of the biology. We now know that these tumors are not the same as adult cancers at all, and treating childhood brain tumors the same way as we treat adult brain tumors is really not sufficient. And the biggest challenge that I sort of see at the moment is I feel that the science is proceeding very rapidly, and every day we're learning more and more things about what causes a childhood brain tumor to form. But a real challenge that we have in oncology is actually how do we translate these findings into the clinical care for our child, right? We can subtype medulloblastomas with really high resolution, and you've seen that, and I'll show you that as well. But how do we actually make that a change into the clinical care of the children, and how do we then hopefully use that to improve survival? And for me, I always sort of think about it, well, you know, the dream that we all have is that when we meet a patient in clinic, we'll be able to say, little Jessica or little Joey has this brain tumor, and we'll be able to tell the mum or dad or the family that their little child has a tumor. We'll be able to give them exactly the drivers of the tumor, and then be able to say, here's a therapeutic approach that will target your child's tumor, and we'll be able to give them a high chance of cure with low toxicity, right? That's what we all are aiming for. But for that to happen, many things need to come into place, right? There are many pieces of a puzzle that I think need to all come into place for us to be able to effectively offer precision medicine approaches for every child in clinic. And so these are very basically some of the things that I think about and what we think about in the lab. We need to learn about, so we need to identify the mutations and the genetic drivers. And I think as a group, the field has come a very long way in trying to understand the biology. But we need to understand what each of these alterations actually do in very deep biology to actually cause the tumors, right? Because if you don't understand what all of these alterations are doing in causing a tumor to form, then how are we ever going to reverse that? So there has to be really deep biology. We need drugs that will be specifically able to target those alterations. And in neuro-oncology, the bar is even higher because not only do we need drugs, we need those drugs to be able to penetrate through the blood-brain barrier with high enough concentration to get into the brain to have an effect. And then assuming we have a good understanding of the biology and we have drugs to target them, we know that tumors are not just a stagnant population of cells. They are heterogeneous tumors with genetic heterogeneity, transcriptional heterogeneity, and the selective pressures placed by treatments, the bottleneck placed by treatments, makes the tumors evolve so that they become resistant. And so we need to preemptively actually understand the resistance mechanisms of all of these new inhibitors rather than waiting to go into clinic and then knowing that the kids are going to become resistant. So this is, to me, a big part of the puzzle. We need to have assays that will accurately diagnose these tumors, and as Dr. Cohen said, in a rapid turnaround time so that they're actually clinically relevant. And then once we all have all of this, we also need effective ways to monitor response. Right now, we heavily base our monitoring on MRIs, but are there other ways to actually track response to these therapies? So it's actually a really high bar to be able to successfully have precision medicine approaches for kids with brain tumors. It's what we're all hoping to get to, but I think we have a really long way to go. But we are learning a lot, and I think, you know, even in the span of my career, which is still evolving, the way, as I said, we've been thinking about tumors has completely changed. So one of the big things for me is this realization that pediatric tumors are not the same as adult cancers at all. So here I'm showing a plot that on the x-axis has different human cancers, and on the y-axis has somatic mutation rates. And one of the big lessons that we have found in pediatric cancers, not just brain tumors, but including brain tumors, is that by and large, childhood cancers are much simpler in a genetic sense compared to adult cancers. So on the list of tumors, I've highlighted the pediatric cancers, and you can see on the right, the tumors that have the highest mutational frequencies are adult cancers, such as melanomas that are associated with environmental exposures, right? So UV light causes mutations that increase the rate of transformation, and so melanomas have high mutation rates. But childhood cancers have really low somatic mutation rates, and some of them, for example, rhabdoid tumors or low-grade gliomas, which I'll show you, and I know Dr. Hawkins will show you later, actually defined by single driver alterations. Now, this is a very unique thing for childhood cancers, because it makes you think that there's something that has gone wrong in development where a single alteration is sufficient to turn on a pathway that is important in development and be sufficient to actually induce tumor formation. So this is another way to look at it. Here I'm showing you copy number profiles of 3 ovarian cancers. So each column here represents a tumor, and I'm showing the different chromosomes. Where you see red, it's an amplification or a gain. So more than the 2 copies of DNA for each of those locations. And where you see a blue, it's a deletion. And when you look at these 3 tumors, you can see there's a lot of red and blue. There's a lot of aneuploidy, a lot of genetic disruption. And it's actually very hard to look at this and say, the driver of this tumor is this gene on whatever chromosome, right? These are just very disrupted tumors. When we look at 3 pediatric low-grade gliomas as a contrast, so here are 3 diffuse astrocytomas that harbor MIVAL1 rearrangements, you can see there's a lot of white, right? So these are very genetically stable tumors. And you can see there's one region where there's an amplification or a structural variant that is sufficient to cause this tumor. So this has some implications when we think about childhood tumors. One is that it has implications for diagnostic studies. So if this child comes to me in clinic and has one single driver alteration, and the diagnostic panels that we use are, for example, designed for adult tumors or don't encompass all of the driver alterations that we see, the likelihood is that we'll miss this driver because it's just this one single event, right? So when we think about diagnostic platforms, they need to be designed specifically for childhood tumors, keeping this in mind. But the other thing is that it actually gives us a lot of therapeutic optimism, because if there's just one driver turning a tumor on, then if we can effectively target that driver, then perhaps we can actually make the tumors respond quicker, right? So an ovarian tumor, if I go in with a drug to target chromosome 7, for example, there's many other alterations that are contributing to that tumor's growth. Whereas in pediatric brain tumors, many of them, particularly the low grades, are characterized by single driver events. As you heard from Dr. Mille, we know that even though histologically the tumors might be called the same entity, that molecularly there are many different subtypes. And this knowledge has evolved over the last decade, starting with some of these early papers in medulloblastoma based on gene expression profiling that showed that there are many different subtypes of medulloblastoma within this overarching group of tumors. And the same patterns are seen across all of the different tumors, where pediatric low-grade gliomas have multiple genetic drivers, high-grade gliomas have multiple genetic drivers, and so there's complexities within each tumor type. We are in the lab across the world working collaboratively to try and develop strategies to target each of these drivers. This really is based on a deep understanding of the profiles of the tumors, the biology that is associated with the genetic drivers, and then understanding vulnerabilities either through genetic assays such as CRISPR screens, compound screens, and really deep cancer biology to try and understand each of these drivers so that we can hopefully have therapeutic approaches to target them. So when I sort of think about what is the role of molecular characterization or profiling, there are multiple roles, but the really two practical things are to aid diagnosis, as Dr. Mille has shown you, and then to see if we can use that to inform therapeutic approaches for the kids. So there are many different ways that you can molecularly profile a brain tumor. At Dana-Farber and Boston Children's, we use a hybrid approach, as do many other centers. And I think this is one of the messages that I want to sort of pass on, is that I don't think that there's one specific genetic panel or profiling method that really gives you the answers all the time. So in our program, and I said in others, we have two DNA-based assays. One on the left is looking for copy number. So it's an array-based assay, which looks for copy number alterations across the genome. But as I just showed you, often pediatric brain tumors have very few copy number alterations. They can have mutations, and they often have mutations in the intron. So we complement the copy number-based assay with a hybrid capture-based sequencing panel, which is called OncoPanel, that is constantly changing, but it basically encompasses about 500 genes that are most commonly mutated in cancer, including all of the exons and the introns that are most commonly associated with cancer. We also frequently perform a RNA-based fusion panel. And then as we've looked to see the ability of these panels to actually be clinically relevant, when we look at the different subtypes, we find that they are actually, if you combined copy number and mutation and a fusion panel, it is sufficient to subtype or detect the alterations in the majority of pediatric brain tumors. I have a couple of sort of points there. The fusion panel and the clinical sequencing panel needs to be designed specifically for pediatric brain tumors. Applying a panel that is designed for adult cancers without taking into consideration the alterations that are most commonly found in brain tumors will miss the alterations that we find in brain tumors. And then, as Dr. Mille has shown you, and you'll hear from Dr. Aldape, the ability to also subtype or cluster tumors based on their methylation and copy number profiles, comparing it to a reference classifier of thousands of of methylation profiles of cancers, has added an additional ability to classify brain tumors based on this. But all of these assays have pros and cons. The methylation profiling is very effective in clustering and telling you that this tumor most closely represents or resembles, for example, a low-grade glioma or a high-grade glioma, but it doesn't give you the mutation cause that you might need to actually be able to say that this child has a mutation that would respond to this drug. So our approach is to complement methylation profiling with a mutational-based profiler like the hybrid capture, and then copy number you can get from methylation or from an array-based copy number profile. So that's a little bit confusing, and I think, as Dr. Cohen pointed out, this is really evolving across centers as to the best way to do this in a clear, certified, efficient way. But I think we also need to understand that there are nuances for each of these, and not one method is 100% effective in classifying every single tumor. And I think we all need to recognize that. You've heard about the version 5 of the WHO, which basically I've just put up. I'm not going to go through this other than to say that it really has increased our understanding of the heterogeneity of each of these subtypes, right? So gliomas are not just low-grade or high-grade. Gliomas encompass many different disease types. Each tumor is a rare tumor in its own right. Similarly, low-grade gliomas are also heterogeneous, right? We've known that they're pathologically heterogeneous for many years, But we also know that they're now genetically heterogeneous. And I'm going to go into low-grade gliomas a little bit more as a primer for Dr. Hawkins' talk a little bit later. Similarly, high-grade gliomas, right? They're not the same as adult IDHs in the majority. They're defined by different histone mutations, and each of these are starting to have different prognostic and therapeutic implications. And similarly with ependymomas and medulloblastomas. But, you know, we are increasingly subtyping and recognizing the subtypes of tumors, and that has implications for how do we treat the kids, how do we design clinical trials, because now we're making a group of rare tumors into a group of very rare tumors. But clinically, we also need to remember that even though that there might be different subtypes, these subtypes can often share things, challenges, treatment approaches that are relevant for multiple subtypes. So I've just put this slide up as an example of infant brain tumors, right? Each of these are rare in their own right, but unfortunately we see them frequently enough in our clinics. Babies have unique challenges in terms of surgery, location of the tumors, the ability to access trials, the ability for children to be able to swallow the drugs, resistance. And so here, understanding the subtypes is important, but also recognizing that sometimes these treatment approaches and the way we think about it is actually, we have to think about the challenges that are common across some of these different tumor types. I've also, at the start I said we need methods to be able to track responsive tumors, and, we all use MRIs. The last 5 years or so, there's been a lot of excitement about the role of liquid biopsies, particularly in non-brain tumors, right? Adult cancers or children with extracranial tumors. And there has definitely been a lot of work across groups to try and understand how relevant liquid biopsies might be for kids with brain tumors. And there's been a few studies that have looked in blood or CSF to try and track, for example, histone mutations, because wouldn't it be wonderful if we could actually diagnose a brain tumor without the need for a craniotomy or be able to actually track this. But brain tumors have extra challenges compared to extracranial tumors. They have a blood-brain barrier that changes the amount of circulating DNA or DNA from the tumors in the blood. And children have extra challenges where it is hard to sample large amounts of blood for liquid biopsies. And so we've been interested in trying to actually understand how reliable these assays could actually be for children. And in particular, we've been interested in actually really defining what is the sensitivity. So if you take 100 kids with brain tumors, how often will we actually be able to robustly detect cell-free DNA? And then how reliable is it? What is the specificity? And so our first attempt to try and understand this, we actually took an approach and said, we're going to try and collect samples from every child that comes into our clinic prospectively. And we designed two genetic platforms to try and detect cell-free DNA. So this is in collaboration with Victor Adelstein, who leads the blood biopsy team at the Broad Institute. And basically, we collected samples from plasma, CSF, if kids had lumbar punctures, and urine, and then designed a pediatric brain tumor-specific hybrid capture panel to detect mutations and fusions, and also applied ultra-low pass whole genome sequencing, and to see if we could first detect copy number or mutation profiles that are suggestive of cell-free DNA, and then compare it back to the matched primary tumor to actually see how reliable these profiles were. And so in this study, we actually ended up collecting samples from more than 250 children, predominantly blood and urine, and a smaller number of CSF. And we included all histological subtypes. the breakdown of the tumors in this cohort match what we see in clinic, where low-grade gliomas are the most common, followed by medulloblastomas and high-grade gliomas, and then some of the other rarer subtypes. And the first thing that we did was collect the blood and the urine and the CSF, and we performed ultra-low-pass whole sequencing, so 10x sequencing of the whole genome in the cell-free DNA extracted from these samples. And we looked at the normal matching tumors and looked to see whether the copy number profiles of what we were detecting in the cell-free DNA matched what we were detecting in the tumors. And the result was eye-opening and disappointing, where we took 260, and the majority of the copy number profiles that were being detected by the ultra-low pass did not actually correlate with the tumors, and only 13 samples from 10 tumors actually had copy number profiles. So this was sort of a first sign of, even though we're excited about the ability to be able to use liquid biopsies, ultra-low pass whole genome sequencing of cell-free is not sufficient. And the reason for this is what I alluded to at the start, is if you look at the copy number profiles of the kids who are coming through clinic and that we're collecting cell-free DNA on, the majority of children have tumors with very small amounts of aneuploidy and have very balanced genomes. So here we're showing a heat map. All the tumors represent all the columns represent a single tumor. We've got the different chromosomes. Red is the amplification, blue is the deletion, right? So you can see there's a group of tumors here on the left that have aneuploidy. And of these, we detected copy number profiles that matched in a subset, so 13 out of 78. The rest of these didn't even have copy number profiles that would ever be able to detect by cell-free DNA. So that's the first sort of lesson from this, was even though this approach is really exciting and successful in things like breast cancer, solid tumors that are extracranial, it's not sufficient for kids with brain tumors. So then we said, well, okay, if we can't detect copy numbers, can we detect the driver alterations? And so here we designed a panel which had 46 of the genes that were most commonly mutated or altered in childhood brain tumors. And we had benchmarked this panel to be able to actually detect tumors, cell-free DNA with at least 80% sensitivity with a tumor fraction of 0.01% with very deep sequencing, so 10,000 X coverage. And we applied this to a subset of the tumors. And similarly, the results are actually really disappointing. So when we first looked at the results, you get your sequencing reads, and we said, here's a child with a low-grade glioma. Can we see the V600E mutation? And so yellow, 2 tumors here where we saw V600E mutations where the tumor had V600Es. So that was kind of exciting. But then we said, well, wait, what is the actual accuracy of this, right? What is the specificity? So we then went and looked at a bunch of other tumors that we knew did not have the V600E mutation and said, can we actually see the mutation in the reads? And sure enough, you could see them, right? So what we found is that if you sequence tumors, if you sequence cell-free DNA from tumors and go in and say, in the reads that come out, can we detect whatever mutation we want? There's a really high rate of false positives because of sequencing artifacts. And if you don't look for that, which a lot of people don't, if you're not aware of this, you might actually say, hey, I can detect the V600E mutation, but you're not actually tracking anything other than noise. We did that across a number of other mutations. So there's been a lot of excitement, for example, in detecting the K27M histone mutation, IDH. And similarly, with just looking at the reads and seeing what the mutations are there are really high rates of false positives. And this for us was, from a clinical point of view, really disappointing, but from a scientific point of view, really important to actually say, people, we need to be careful about this, because right now these assays as they stand, if you use these sorts of approaches, are not sufficient to meet a clinical metric. So we tried to then apply algorithms which would try to reduce the rates of false positives. And one of the ways that we did that was to actually apply a unique molecular identifier pipeline. so that you can actually track the sequencing reads to which DNA molecule they came from. And when we use these more stringent approaches, out of the 86 samples that we had applied to this deep sequencing, we were only able to detect cell-free DNA reliably and robustly in 4 of the patients. And all 4 of these were from high-grade gliomas. And so this was a study that I think is important in showing us that we still have a long way to go in trying to be able to apply liquid biopsy approaches for kids with brain tumors. We think there are a number of reasons why it's harder to do this than it is in some of the other extracranial tumors. One is, as I said, in childhood brain tumors and other childhood cancers, there are fewer numbers of somatic alterations to detect. There could be technical reasons, and I'll show you that it wasn't because of our coverage, but we think that the rates of circulating tumor DNA is low to start with. And then in a small child, we're limited by amounts of sample that we can actually obtain. So first question was, of the tumors that we profiled, how frequently did they actually have an alteration that we would have detected with that panel? So even though we prospectively designed the panel and said this should detect the alterations in the majority, in real life, when we applied the panel to kids coming into clinic without selecting out anything, the majority of children actually only had 1 or less alterations, right? So we weren't even able to detect the alterations in the majority of children. We were able to go in and look to see what our coverage was. So of the tumors that had callable alterations, when we actually looked at the number of reads that we're getting more than 1,000x coverage. So that was above our sensitivity. So we were able to achieve enough coverage. But this was the big reason that we think it's really challenging, is that we know that at 1% cell-free DNA concentration or circulating tumor DNA, our assay has 90% power to detect alterations in 80%. But we only detected alterations in 4%, right? So 4% of our reads, so 1% is this blue line. Our panel was powered, or is sufficient to actually detect alterations more than that, but we only detected samples in 1 of these. So our conclusion here is that the main reason why we're not detecting cell-free DNA in these kids is that even though the panel is able to detect low concentrations, the samples themselves that we're obtaining from the kids do not have enough cell-free DNA to actually detect it. So I just wanted to put that out there because there's been a lot of excitement, and I think we need caution and we need more methods and technologies to really increase our ability to detect cell-free DNA. I just want to bring it back to how is all of this actually playing into our clinic and when we're seeing children who are coming to us with brain tumors. So I'm going to use low-grade gliomas as an example of the advantages and the benefits. And the reason for this is that I think that over the last 10 years, low-grade gliomas has become a poster child for targeted medicine and precision medicine approaches for pediatric cancers in general, but especially brain tumors. I always talk about low-grade gliomas in kids and make this point that pediatric low-grade gliomas are very, very different to adult low-grade gliomas. They have very different drivers, but they also have a very distinct clinical course that we need to keep in mind as we start using these new inhibitors. So unlike an adult patient with an IDH mutant low-grade gliomas, the rate of transformation of pediatric low-grade gliomas to a higher grade tumor is very low. So this was a study that we did approaching 10 years ago now, where we looked at the SEER database. So the SEER database is an American, a US database, cancer registry, where you can track diagnoses, but also some limited treatments, whether they had full surgical resections, subtotal resections, and then outcomes. And what we found is that when we tracked 4,000 children who were diagnosed over the decades before, the majority of kids are still alive as young adults, whereas an adult chart, sort of they progress and die within 10, 15 years. We've also learned that these tumors are genetically heterogeneous. This is a commute plot of the genetic alterations found in pediatric low-grade glioma. So again, every column represents a tumor. And the genes here that we're showing you on the left are the genes that are most commonly alterated. Here, this was a cohort of about 150 children that we profiled with whole genome sequencing. There are a couple of messages that I want to make, want you guys to take from this commute plot. First is that I've color-coded the alteration. So where you see orange, it's a structural variant. Where you see blue, it's a point mutation. So low-grade gliomas, by and large, are a disease of structural variants and fusions versus point mutations. And the most commonly altered gene is BRAF, as you know and as you've heard. But then you also see mutations in other genes, most commonly converging on the MAPK pathway, for example, FGFR, and we're starting to find more and more FGFR alterations as we biopsy more of the midline gliomas. And then a subset of gliomas, as you also heard, have MIB alterations. The other message, which I will keep going back to, is that you'll see that there's a lot of white, right? So these are single driver alterations. But we were, as a community, lucky in that the most common alterations for kids with low-grade gliomas converged on the MAPK and mTOR signaling pathways. And the reason I say we were lucky is that the same pathway, as you know, is mutated in melanoma. And before the discovery of these alterations were made in low-grade gliomas, our adult colleagues working with Farber had already developed and tried a number of drugs that targeted this pathway, including BRAF and MEK inhibitors. So one of the biggest challenges that we have for targeted therapies of precision medicine is actually having drugs that work and having drugs that we can use in children. And we were able to leverage what our adult colleagues had learned in melanoma for kids with low-grade gliomas. And this has allowed a number of different trials to actually target these acts, the MAPK or the mTOR, and we've had a number of clinical trials now studying the efficacy. So there are type 1 and type 2 inhibitors that can directly inhibit BRAF. There are MEK inhibitors that will target the pathway downstream of BRAF. And then there are drugs such as everolimus that can target, for example, the mTOR pathway. And all of these drugs have gone through varying stages and are going through various stages of clinical trials for children with low-grade gliomas. I put this slide up because I think it's really important for people that do not think of genomics frequently to understand that a BRAF mutation is not the same as a BRAF rearrangement. We often use different words to describe a BRAF rearrangement. You'll hear them talk about a paired limb duplication. You'll hear us talk about Ki-1549 BRAF. You'll hear the words BRAF fusions. This is really important because there are major therapeutic implications if we don't understand the differences. So the BRAF V600E mutation is a point mutation that results in constitutive activation of the BRAF kinase. In the BRAF rearrangements, we have a juxtaposition of another gene. Most commonly, it's Ki-1549, but there can be multiple partners. And this juxtaposition results in activation of BRAF by loss of the negative regulatory domain. Right? But this fusion can still dimerize, and you still have a wild-type allele of the BRAF that can also dimerize and function. So as these mutations were discovered, the two different BRAF inhibitors have gone through trials. the type 1 BRAF inhibitors that were developed for melanoma that target the V600E mutation have been really exciting and have shown a lot of efficacy in children with that BRAF mutated or that V600E mutant low grade. So here's an example of a patient that we treated, had a large tumor here, had a debulking biopsy. We don't want to, result in neurological deficit. So the surgeon appropriately left residual here, and we treated this child with a type 1 BRAF inhibitor and saw really unprecedented responses. It's very uncommon for us to see such degrees of responses with vincristine carbo or some of the traditional chemotherapies. So this has been really exciting. However, if you use that same drug in a child with a BRAF rearranged glioma, then we can actually cause some harm. So this is preclinical work that shows that if you have cells that have the V600E mutation or the BRAF rearrangement, and you treat these cells with a BRAF inhibitor, you can see here on this Western blot, I'm showing activation of ERK, which is one of the substrates of the MAPK pathway. And you can see that the type 1 BRAF inhibitor really effectively shuts down this pathway. And that's why we're seeing the responses. However, in children with the BRAF rearrangements, if you use these type 1 BRAF inhibitors, not only do you not get the same level of suppression, with prolonged dosing and high dosing, you actually get increased activation of the pathway. You get a paradoxical activation and growth of BRAF rearranged gliomas with a type 1 BRAF inhibitor. And that is because you still have a wild type BRAF allele there. It's still able to dimerize and result in paradoxical activation is what we think. So I think this is really important for people when you start getting the genetic results back to understand the differences between the mutation and the rearrangement and the fact that type 1 BRAF inhibitors are contradicted. The good news is that there are other ways to target this pathway for children with the BRAF duplicated low-grade gliomas, and MEK inhibitors have got through phase 1 and phase 2 and are now being evaluated in phase 3 studies for these children. And the results have been pretty exciting. And again, very different in terms of responses to what we commonly see with traditional chemo. So selumetinib is one of the MEK inhibitors. This waterfall plot I'm showing you is from the Pediatric Brain Tumor Consortium study led by Jason Fangasaro, where in a phase 1 and phase 2, they treated a cohort of kids with low-grade gliomas. Here you can see a number of children actually had reductions in their tumors, including BRAF fusions or rearranged in blue and mutated. There are some challenges. As you can see, there are 4 children here where the tumors didn't respond, and we need to understand why. There are a new group of BRAF inhibitors. You'll hear them described as type 2 BRAF inhibitors that are now being evaluated as a way to stabilize BRAF for the kids with rearrangements. That drug or those drugs are going through early phase clinical studies. But right now, if you have a BRAF rearranged tumor, the most tested way to target it is through the use of a MEK inhibitor. But there are major challenges, I think, emerging that we are behind in the lab in trying to understand. And I think this actually has pretty major implications in that in the children's oncology group right now, we have 2 phase 3 studies which include MEK inhibitors, one for children at diagnosis and one for children at relapse. And what we are starting to find, not in that study, but just in kids that have been treated, is that we see impressive responses in a lot of children. So this is a child this was actually a V600E ganglioglioma that we treated with a type 1 BRAF inhibitor here. Got a pretty impressive response with 12 months of treatment, but when we stopped the drug within a month, the tumor has grown back. And we're starting to see this pattern of regrowth or rebound when we stop the inhibitors for both BRAF and MEK inhibitors in more than 50% of children. We're still trying to work out what the real numbers are because this is happening in real terms. But now we have had kids who have been on these inhibitors for 4 or 5 years continuously versus what we would have done in the past, which would have been 12 months of vincristine and carboplatin. And for me, I think biologically, what I'm concerned about is we do not have any understanding of the long-term implications and side effects of the use of these drugs for children. We're going to learn that in the next 10 years. And we don't know if this is going to play in with that natural history of stabilization of low-grade gliomas. And so this is something that we need to understand. And then, as I said, we still don't have a good understanding of why some children don't respond to these to these drugs. So this is just my last slide, and we can have a little bit of time for discussion. But as you heard in Dr. Miller's talk and as we're seeing in the lab, I'm really excited by the biology that we have learned and are continuing to learn over the last 10, 20 years. I feel like we are living in the era that our leukemia colleagues did in the 70s and 80s when they took an incurable disease and were able to cure leukemia. I feel that we're living that right now for children with brain tumors, and it's really exciting to think where we're going to be in another 10 years. But we also need to be, I think, cognizant that we don't know what we don't know, and we have no idea about how best to use these drugs, how to overcome resistance, and to be really cognizant of the fact that we are going to learn about the long-term effects of these drugs as they come, right? We need to understand the biology. I think the BRAF story is a big one in terms of if you don't understand the biology and just try and use an inhibitor for a mutation and alteration in a gene, that we can do harm if we don't actually understand the biology. And then finally, just wanted to thank everyone in my lab, but also one of the joys of doing what we do is working collaboratively with people across the world. And it's really exciting to think about what everyone is going to hopefully achieve together for these children. So thank you again. And I'm happy to take any questions if you have any.
Thank you very much for an excellent talk. I'm looking in the chat. I see some thank yous, but I don't see a question. So let me start off with a couple of comments. As I said, this is an excellent talk. To get to the basics here, so what you've shown the group here is that we can analyze tumors, looking at the tumor tissue, leaving aside the cell-free DNA question for a moment, with a variety of technologies now. There's next generation sequencing, which is a generic term that now encompasses a lot of different approaches. And one can do targeted next generation sequencing where you know specific genes you want to look for, or there's whole genome sequencing, which is a different enterprise entirely. And there are different approaches to this. There are different costs to this. And so we're looking at various ways that are getting that done. There's also the issue of chromosomal aberrations that are at a chromosomal level rather than at a specific gene level. And those are detected by different technologies, as you've shown. And so our clinical colleagues and others across the world need to know that as we're looking at these, there are multiple ways of classifying that are complementary rather than I'm going to do this one or I'm going to do that one. I think there's a lot of impression of let's just do NGS, and that doesn't work, as you pointed out, very effectively. It doesn't work in all of oncology very well, but it certainly works even less well in neuro-oncology and pediatric neuro-oncology. And then finally, there's the methylation array issue, which will be coming up shortly from Dr. Aldape, and which we still don't quite know how much trust to put in that, to be honest. So there's a single database which is constantly being added to. But the questions that I have, and you can direct your attention to this, is Ken will also, obviously, but what is the gold standard for diagnosis when you have a cluster what do you call that cluster other than just saying it clusters? Where are the gold standards? Can we take a tumor that somebody called pilocytic and say, no, it clusters with gangliogliomas or whatever? Go ahead.
Yeah, I mean, that's an excellent question. And I think as a community, we really need to understand these nuances, because the reason I say this is, sorry, I think someone else is, I think there's a danger in, as you said, saying, and we see this in clinic already, where we get a patient come in and they say the tumor has been profiled and there's nothing identifiable. But unless you actually understand how the tumor has been profiled and understand the nuances, you can't interpret that. So my feeling is that it's not one thing. The histopathology is never going to be made redundant by genomics, right? Pathology and what the tumor looks like is a really important part of the diagnosis. And then whether it's a mutation or a copy number profile or a methylation, these are just different ways to look at the fingerprint. And you're looking at that fingerprint of that tumor from different angles, right? One is either with DNA and mutation, one is by methylation. And so, to me, the gold standard is an integrated diagnosis that combines the histopathological interpretation, looks at the mutational profiles and the copy number profiles, and looks at the methylation classifier and says, when I look at these three different metrics, this tumor is most consistent with whatever. Because as you said, the methylation classifier will just tell you what tumor that tumor is the most close to and clusters to. It doesn't tell you anything about whether it's a BRAF V600E mutation or a higher rearrangement. It just tells you what that methylation profile most closely resembles. So in my opinion, a methylation profile by itself is not sufficient. I think you need that mutation call. I think you need the copy number calls, which you can get from methylation or copy number. I think in pediatric brain tumors, where we're frequently seeing single structural variants and rearrangements, that often you need an RNA-based fusion caller to complement it. So there are nuances, and these sorts of interpretations and diagnoses, in my opinion, need to be done in centers that are looking at childhood brain tumors all the time.
Thank you. As a neuropathologist, it's nice to know I'm still going to have a job for a while.
Oh, you have the most important job.
Can I ask a question, Dr. Mille?
Of course, please.
Thanks, thank you. If you have covered it, I apologize, but when we do pathological biopsies in a tumor, sometimes, depending where you are taking the biopsy, you might get a different answer. And in the same thing with these molecular pathology diagnosis, are you going to get a uniclonal tumors, or are you going to have tumors with different clones, and are they going to affect how you are able to apply when it comes to treatment?
You want me to take that, Dr. Miller?
Sure, yes.
We have looked at that, as have others, right? So it's a really good question, and I think the answer is we need to look at it more. But the basic drivers that we are studying, right, the BRAF mutations or the histone mutations, that they tend to be clonal, and therefore present at all of the different parts of the tumors. When we have, in the small number of tumors that we've looked at with the multi-regional, so the drivers tend to be clonal. What I think is an unanswered question is metastases, right? Primary and METs, and how similar or different they are. I suspect there'll be a clonal trunk. The truncal mutations will be similar, but whether there are sub-regions that have additional subclonal mutations possible, I don't think we've, as a community, looked at enough tumors. And then the other big thing from a surgical perspective that we talk about a lot is the role of rebiopsying. on progression, right? We have a tendency not wanting to put our children through multiple craniotomies, but particularly as we start using these targeted inhibitors, understanding the evolution and whether the tumors are acquiring resistance drivers is something that we're going to need to understand.
Thanks. Thank you.
To add to that, we don't know a whole lot about this in pediatric tumors because they're less common and they're less well studied consequently in this manner. But we do know that for adult gliomas, there's regional heterogeneity. So there are trunk driver mutations that are the main clone that starts the tumor. But then in different regions, you can have subclones that have additional mutations that are different from other regions. And that does complicate the therapeutic process with recurrences. The other thing we haven't gotten into at all here, because it's become less fashionable to talk about it, although it hasn't gone away, is the stem cell theory of cancer as applied to these tumors. And the fact that after you even had a gross total resection, is there a reservoir in a niche somewhere of stem cell white cells, whether they are originally stem cells or otherwise, that have acquired stem cell white properties that can then repopulate the tumor. And these are the cells that are commonly thought to be the resistant cells to therapy. Any comments on that?
Yeah, it is. We're definitely thinking about it a lot more in pediatrics, with the for two reasons. One is the understanding or the increasing understanding that a lot of the mutations that we see are locking the tumors into a more undifferentiated state, right? For example, the histone mutations are locking the cells into a more of a OPC-like primitive state. And then definitely for resistance, I can talk to what my lab's doing. We are studying resistance mechanisms to a number of different drugs, and we definitely see, in addition to genetic mutations causing resistance, that changes in cell state can also, right? The cells will become either a little bit more differentiated, often a little bit more differentiated, depending on the drug, to persist the drug that's been given, and then that can then also cause tumor growth. So yeah, that is very understudied in pediatrics.
And one last point about that is that the things that are done to treat tumors also cause genetic alterations. So it's particularly radiation. Again, looking at an adult model, David Lewis showed years ago that if you looked at IDH1 mutant gliomas in adults that then recurred after radiation and temozolomide, that they had gone from a status where they were not microsatellite unstable to ones that had microsatellite instability and had mismatch repair. And it started to acquire all sorts of new mutations because of that new status. And it was a change from the original tumor. So yes, rebiopsy is important because there will be genetic changes that are therapy induced. Okay.
Dr. Mille, can I just ask one quick question, if I may? Pediatric tumors are not that many to start off with compared to adult tumors. And now that we had the histological classification, that makes subgroups smaller, but now we have got so many genetic aspects to look at. How do we then have enough tumors to do studies if there are so many heterogeneity? Any thoughts, Dr. Miller as well as Dr. Bandopadhayay? Yeah.
Cancer is obviously collaboration in large consortia. So in North America, we have the Children's Oncology Group. In Europe, there's an equivalent group. It's SIOP. And we are now, of course, extending these international collaborations. So COG includes a bunch of Australian institutions. I think it's important that we pool data, we pool specimens in common laboratories that do these kinds of investigations, because otherwise single institutions will never come to answers on these very rare tumors. You're right.
Thank you.
A question in the chat, which is, what is the bottom line message here? And we're going to run short on time, but I think the bottom line message is that the landscape has changed. We no longer just take something out, look at the pathology, give a diagnosis, and have a cookbook treatment. And so treatments will become more and more personalized as the pharmaceutical industry develops targeted therapies for the particular identified genetic alterations that are there. And so that's the lesson. We're not there yet very much, but we're getting there.
Yeah, and I'll just add to that message, because for me, this has been a big lesson, is that BRAF is understanding the biology, right? Just saying that there's a mutation in the gene, therefore the inhibitor will work, is dangerous, right? So you absolutely need to understand the biology and the nuances of the inhibitors that are being used before we can just use them.
I think just a small mention that Mr. Salindra was wondering what's the bottom line for countries with developing countries where they can't afford these all these genetic tests. I suppose here the maximum resection, and then able to see what can be afforded. Any comments on that, Dr. Mille, Dr.
Well, there are two aspects to this, Naren, that I think are relevant. One is availability, and that in terms of geography is now really not an issue very much. There's FedEx, and most of this stuff can be done in paraffin and fixed tissues, fixed in paraffin embedded tissues. So if you have the resources to send it to somebody who can do it, whether it's here in the States or in the United Kingdom or in Germany or wherever, then I think that that's something that can be done. The question, though, is the other question is cost. These are not cheap tests. For us here to send an adult glioma out for both chromosomal array and NGS testing is in the vicinity of $6,000 at least, that somebody's going to pay. There are companies like Foundation One or Caris who are right now doing this at almost no cost to patients, because either Medicare or the insurance companies will cover this for adults. But for pediatric tumors, that kind of coverage is less available. And then in the international setting, for countries that don't have it in-house, those costs can be prohibitive. So I think the international community may have a responsibility, just as they've talked about the responsibility for getting vaccines for COVID out to places that don't have them otherwise, that we need to find ways to do this for our pediatric patients across the world.
Thank you.
I think we should probably
Sure.
move on. I don't know that Dr. Aldape.
Yes, Dr. Aldape is here. Thanks.
Very good. Then let me introduce him, because he's going to give the next talk. So Ken Aldape is a molecular neuropathologist. He is currently the chief of the laboratory of pathology at the NCI Center for Cancer Research. Dr. Aldape earned his MD from the University of California, San Francisco, and then he did his residency and fellowship in anatomic pathology and neuropathology there as well. He was appointed to the faculty at UCSF and was there for some years, and then was recruited on to MD Anderson Cancer Center, where he ultimately was chair of the Department of Pathology. He then moved to Toronto and was at Toronto General Hospital and Research Institute, and had a clinical appointment at the Princess Margaret Cancer Center. And then he was attracted to move to NCI. He is doing a considerable amount of work for as a central referral laboratory with methylation array classifiers, as well as other kinds of molecular biology of cancer in general, and pediatric cancer or pediatric neuro-oncology in particular. So I'm going to turn things over to Ken, and I'm going to have to log off because I have a conflict here at the local institution, but I'll be back later.
Thank
Ken?
you. I'll take over. Thank you.
Thanks so much, Doug, for the kind introduction. And I appreciate the opportunity to share what we've been doing on methylation profiling of CNS tumors. I wonder, can you see my slide?
Yes, see it clearly. Yeah, thank you.
Wonderful, wonderful. Okay, I'm going to talk a little bit about our experience using methylation profiling as an adjunct to diagnosis and classification of central nervous system tumors, making the overall point that it's an extremely useful adjunct when placed in the context of the entire case. It doesn't replace what we have, it adds to what we have and adds new value to that to improve accuracy and precision of brain tumor diagnostics. So let's see if I can, there we go, no disclosures. Okay, so classification, I think, as we know it, began about 100 years ago with actually a neurosurgeon, Harvey Cushing, working with a neuropathologist. Also had an initial classification of neuroepithelial tumors And some of these entities are maintained to this day: medulloblastoma, glioblastoma, ependymoma, oligodendroglioma, et cetera. So this has been tried and true to look at the tissue histologically and to diagnose it based on its appearance under the microscope. And that's really the process that's been going on over the 20th century, which was using the microscope as the mainstay of diagnosis of CNS tumors. And although we get reasonably far that way, the problem is that for difficult cases, and I would say it's magnified for pediatric CNS tumors because of their heterogeneity and their rarity, that it can be, in certain cases, subjective. There can be ambiguous cases that are difficult to classify based on how they look. And that ends up being that sometimes we're not able to give a definitive diagnosis. So we give what we call a descriptive diagnosis: high-grade glioma or embryonal tumor or what have you. And you can imagine in that situation, and here's a paper from about 20 years ago, basically looking at histopathology-based diagnostics. And in this paper, and there's many of these kinds of papers looking at 500 cases that were reviewed, that were received from the outside institutions to MD Anderson Cancer Center. You can see that a good proportion, 40% of these cases, were deemed discrepant between the outside institution and MD Anderson. Now, who knows who's correct? I'm not saying that one's correct and the other's not. It's more that there are discrepancies when one uses histopathology as the sole means of diagnosis. So it's good, it's not perfect, and there's room for improvement. The WHO classification, which, as you know, has just come out in the 2021 version, I think, as was mentioned, it increases the number of distinct entities, number one. But also the other advance is that many of the entities in the past were defined by how they looked in the microscope. That's still included, but now you can see, and this is just a snapshot of some of the many entities in the WHO, you can see just by their names that they involve a molecular signature or a molecular profile or a mutation as part of the definition of that entity. So medulloblastomas are broken up into subgroups based on the activation of certain pathways and mutations. Other embryonal tumors, you can see here just scanning down, are defined by the presence of a molecular alteration, such that the molecular alteration is required for that diagnosis. So no molecular alteration, no definitive diagnosis. So diffuse astrocytoma, MIB or MIB-L1 altered, requires the establishment of alteration in this gene to make that a definitive diagnosis. So that, in concept, will reduce subjectivity and increase accuracy and precision. That said, we do, as pathologists, hold to the tried and true method of an integrated diagnosis. There's no one kind of modality that can be used in isolation. We need all the information we can get, and we'd like it to all fit together into a pattern that gives us an integrated diagnosis. And that includes the H&E, that includes the radiologic imaging, molecular testing, if available, the clinical history of the patient, age of the patient, location of the tumor, and clinical history, as well as some special stains that may or may not be relevant. And so all these fit together. And here's just an example of ETMR, embryonal tumor with multilayered rosettes, histology showing a typical pattern, immunohistochemistry of a particular stain, Lin28, imaging is consistent. The clinical information is consistent, it occurs in a child, and then the use of things like next generation sequencing or FISH to find the C19MC amplifications present in some of these tumors. They all fit together for an integrated diagnosis, such that no one of these modalities by itself can give us a diagnosis. We need it all to fit together. And so, as I mentioned, there is a lot of heterogeneity in these brain tumors, and there's case-to-case variation on the importance of some of these modalities. In some tumor types, the H&E is nearly diagnostic without any issues. There's other cases where the H&E is just the beginning. There's really not very specific for a specific diagnosis. It may be that the molecular is critical. So there's case-to-case variation on how these data are integrated. So, as you know, some tumor types have characteristic features. Medulloblastoma is required to be in the posterior fossa, as you know, typically in children and young adults. Other tumor types have highly characteristic histopathology, for example, subependymomas. Other types have a wide range of histopathology, but are unified by a single test or just a small number of tests. For example, ATRT showing loss of INI1. In the real world, we find that many cases don't fit perfectly together into a recognized entity. Sometimes it doesn't all fit into something that we can be confident of. For example, the case may lack characteristic histopathology, or perhaps it has histopathology that's suggestive of a particular tumor type, but it's an unusual clinical setting. Maybe the patient age is not typical, or the location, etc. And so when things don't fit, as I mentioned, then that's where it leads to inter-observer variation and diagnoses that are what we call descriptive. They're not specific for a single entity, but they're more general. And so what we're talking about today then in this talk is how methylation profiling can add to that. Not to replace all these data sources that are critical, but how it can supplement things when we can't get to a definitive diagnosis by conventional means. So the methylation pattern, from a simple point of view, represents the cell of origin. And that's one basis of how we classify tumors, is based on the presumed cell of origin. And layered on top of that are the changes that occur epigenetically that are acquired during tumor genesis. Some changes that occur during the formation of a particular tumor type can have a profound effect on the methylation pattern, and we can use that to our advantage. And so, in addition, even when histopathology is definitive, like for example, medulloblastoma, we can refine that by methylation pattern into one of its subclasses. An advantage here of methylation is it's a stable marker of the cell. It's stable over time. When we take matched primary and recurrent tumors, we find that the methylation pattern is quite stable over time. And so that gives us a good sense of stability for the use of methylation. It works well in the samples that we have to work with, which is formalin-fixed paraffin-embedded samples. That's another practical advantage. And it's a single test that can be implemented, that it can be applied to a wide range of tumor types. We have many immunohistochemical stains. Many of them are specific only for one or two tumor types, and they all have to be individually validated, et cetera. This is one test that in concept can be used for a wide range of tumor types. So what we know about methylation is that, as I mentioned, it's stable, and it's a mark of cell of origin. And in tumors, it's also stable, as opposed to some other genomic aberrations, for example, gene expression. And so, as I mentioned, methylation profiles are that combination of cell of origin plus the changes acquired during tumor genesis. So here's some normal histology. And if you can imagine, these all came from the same patient. You can ask, well, they all have the same DNA sequence, pretty much, ACTG, et cetera, for 3 billion base pairs. How are these tissues so different? They're not different based on their primary DNA sequence. They're different. The reason, the biologic basis for these differences is epigenetics. So these tissues, here's liver, here's colon, here's kidney, skin, et cetera, differ not on their DNA sequence, but on the modifications on the DNA, and that's DNA methylation. So if we could reverse engineer a situation where we knew the methylation pattern of liver, and we didn't know what the tissue was, we could profile it in concept and show that, in fact, that was liver. So that's the concept behind methylation profiling as a diagnostic. So I mentioned the WHO 2022 Blue Book. It lists a large variety of entities that occur in and around the central nervous system. On my count, it's 110 distinct diagnostic entities that are in just the brain tumor, the CNS tumor blue book. And 22 of these are new compared to 6 years ago. So it's highly complex. There's a lot to keep track of, and it's evolving over time. And going into that blue book, what's new as compared to 2016 is that many of these entities, approximately half to my count, and most of the new 22 new entities have methylation-based classification as either an essential criteria or recommended criteria for diagnosis, showing the value and the probability that the use of methylation is going to increase in our field. It has some advantages in the diagnostic process. With typical methods like immunohistochemistry or FISH, we need to sort of think of the diagnosis and do a test that can either prove it or refute it. So if we think it might be a K27M mutant glioma, diffuse midline glioma, we then need to think of getting that immunostain for K27M or getting that sequencing for the H3 K27 mutation. Methylation has an advantage that you can run it, and even if you didn't think of this, you still can get that suggestion. So it has that advantage that it doesn't require prior consideration of a diagnosis, and therefore can serve as a nice checklist function for diagnosis in case that diagnosis was missed or it wasn't thought of, et cetera. And so I think I've already mentioned this, but the bottom line is that by screening many methylation sites, there are millions of methylation sites across the genome, and by interrogating a large subset of those sites, one can then reverse engineer using machine learning to define methylation classes that track with specific entities. And initially, it was reassuring that many of these methylation patterns did, in fact, track to what we already knew about histologic tumor types: glioblastoma, medulloblastoma, ependymoma. And so that there was this correlation initially between the methylation profile and the histopathologic entity. So that gave encouragement to develop a brain tumor classifier. And that was published now 4 years ago with a large group of individuals, primarily spearheaded by the DKFZ in Heidelberg, Germany. I was one of the middle authors here. As you can see, it was very much of a team effort, but spearheaded by the DKFZ. And here's a figure from that paper showing the initial aspects of the initial classifier. Here there are 82 different tumor entities, each one given a name or abbreviation that you can recognize. Here, GBM, these are some of the GBM subclasses, glioblastoma, and their subclasses. This is diffuse midline glioma, K27 altered. Here's medulloblastoma, sonic hedgehog subtypes. Here's medulloblastoma group 3 and group 4, et cetera, et cetera. So the idea here is this becomes the reference set, that when you now have a new tumor, you profile on the methylation and you see where it goes. So the methylation depends on comparison of your tumor, of the tumor in question, to a reference set. And the methylation classification is only as good as the reference set. So if the tumor entity under question is something that is not in the reference set, methylation will not necessarily be helpful. It could lead to new discovery of new entities, et cetera, but it really depends on having a large and diverse reference set. And, I just showed this case early on from a number of years ago that was important to me, that although, it sounds perhaps not so exciting to do methylation on tumors, but once you start seeing tumors and how it can affect specific cases, you become a believer. At least that's my interpretation. Here's a case that we received on the outside that was given a diagnosis of glioblastoma, and it had some features of glioblastoma, including microvascular proliferation. The patient was getting treatment for glioblastoma, radiation, chemotherapy, et cetera. We looked at it histologically. We didn't think it was, but then it becomes this problem of inter-observer variability and who's right. us versus them. No one really knows for sure because there's no objective basis for that. We ran methylation and we found it to be a pilocytic astrocytoma, a grade 1 tumor, which of course is not something that normally needs such intensive therapy. And we were able to affect the management of this patient. So the bottom line is that it serves as a nice quality check for difficult diagnosis. So what we've done since at the NCI, we started this process about 3 years ago of implementing this clinically for clinical diagnosis. And as Dr. Miller mentioned, we've been receiving cases from outside institutions, they're difficult cases, and we've been to date profiled about 3,000 tumors. And we simply have a practice of any case received from the outside or internally received from our operating room is submitted for DNA methylation, in part to learn how we can use the classifier, and also to, occasionally, even when we think we know the diagnosis by histology, every once in a while we can't be surprised. And so we're using this in the routine diagnostic workup, and we're using the classifier that I showed that was developed from the group in Heidelberg. And we use alternative visualization techniques that I'll show you to complement the specific classifier. So it's conceptually very straightforward. We use tissue unstained slides or tissue block, and we isolate the DNA and we run it on an array, an Illumina methylation array. We always use the same array, and most of the, in fact, others use the exact same array, and that provides a nice sense of uniformity. And that's run on the on a particular instrument, and then we get image files from that, and then that's by using machine learning, as I say, is dissected down into a specific class. With the classifier, one gets a tumor class, medulloblastoma, group 3, ependymoma, Rayleigh fusion, et cetera, et cetera. So you get the class, the name of that, and then you also get a score, a confidence score. The score can range from 0 to 1. One wants the score to be as close to 1 as possible, because that means very, very high confidence. And then clinically, we need to set a cutoff. And so we've used the cutoff of 0.84, just that seemed a reasonable cutoff based on what others had been using. And so for definitive diagnosis, we use a class for a cutoff of 0.84 or above. We oftentimes do not get a class with that confidence score. Sometimes we get what we call a no match. It doesn't match to anything. Other times we get a match, but with a confidence score of below 0.84. Sometimes it's close, 0.8. Sometimes it's not so close, 0.3, et cetera. And so that's when judgment, that's when looking at other aspects of the case become very important. And so, some general rules is that if it's 0.5 or between 0.5 and 0.83, then we consider that suggestive, but it's cautious. If it's below 0.3, then we essentially consider that a no match. And so we've looked at this now retrospectively on the cases that we profiled to date, and we can categorize. We tried to estimate the value of the classifier. It looks great, et cetera. It's pretty fancy, but what does it actually contribute? If it's not contributing anything over and above what we can already do, then why do it? And so we tried to measure that impact by comparing cases that we received from the outside that had an outside diagnosis, pre-methylation diagnosis, and we ran methylation. And then we gave it now a integrated diagnosis that included DNA methylation, and then categorized for what change was made, if any. Sometimes the diagnosis was simply confirmed. It was the methylation simply showed that yes, it was what we thought it was before we did the methylation. Other times it was consistent with the diagnosis, but it refined it. For example, the initial diagnosis might have been medulloblastoma, but methylation was able to show, okay, yes, it is medulloblastoma, but it's a sonic hedgehog subtype, or it's a Wnt subtype. Sometimes, so in that sense, it had a contribution, although it didn't change the diagnosis per se. Sometimes it did change the diagnosis. perhaps that initial diagnosis was not known. Maybe it was descriptive. Maybe it could only be called an embryonal tumor or high-grade glioma. And at times, methylation was contributory to actually defining that diagnosis for that case. Sometimes it was misleading or non-contributory. Maybe it was no match case, or maybe it gave us a low score for some entity that really wasn't probably consistent. So these kinds of things all could happen. So we tried to measure that. And we, as I showed earlier, we used a reference set. We've expanded our reference set now to having over 12,000. This is an old slide. We have now over 15,000 brain tumors that have been mapped on by methylation onto a reference set, and you can see the variety of the brain tumors, et cetera. And so when we get a new tumor, we map it to this, and we find that the higher the sample size, the better we can do with respect to accurate classification. And so, as I mentioned, it requires a large data set. We use that 0.84 threshold, and sometimes we don't get a match. And so we have to use orthogonal data, clinical information, how it looks under the microscope. Are there mutations that are tumor specific? Are there copy number changes that are tumor specific that can contribute to the diagnosis? And what we find is that it's best, and I think it was mentioned earlier, that it's working together, combining cases from multiple centers. We've been starting to work with a number of centers. Really, is going to be essential to making this work, because no one institution is going to have sufficient number of cases, especially rare entities, to be able to pull this off. And so how can we change or refine a diagnosis? I'll give you a couple examples here. Here was one case. It was a high-grade glioma in a young adult patient. It was IDH wild type. And it was near glioblastoma, but it wasn't quite there in glioblastoma. And what we noticed is when we looked to where this particular tumor mapped to tumors that had been previously known to have mismatch repair deficiency, which can occur in children and young adults with high-grade gliomas. And we know that the conventional therapy is contraindicated in that setting. And we were able to provide some information that this patient might respond to an immune checkpoint inhibitor. So methylation can contribute to actually clinical management of that patient by giving an accurate diagnosis. And based on this finding on the methylation, we then were triggered to look for mismatch repair deficiency, and we were able to confirm that. And so that's, I think, how methylation can contribute. It sets up the hypothesis that then can be tested with an orthogonal test and potentially affect patient management. Here's another case that it was a very young child with an embryonal tumor. It was intact for INI1, which is the marker we use most commonly to diagnose ATRTs, which lose INI1. But in this case, INI1 was intact. So what do we do? Well, it mapped to ATRT by methylation, but rarely these tumors can have an alternative mutation in SMARCA4. And so we were able to then prove that. But we wouldn't have thought of doing that if we didn't have that methylation class. So that's an example of how methylation can work together to really integrate a particular diagnosis. And so I mentioned that we measured the impact of methylation in the work that we've done. When we got to about 1,000 cases, we simply asked, what is the contribution of methylation by comparing the pre-classifier, the pre-methylation diagnosis, to the final diagnosis, and what was the impact? And so I won't go over it. I'll just go over the impact here with you at a high level, but the bottom line is that of the 1,000 or so cases, about 2/3 of them, 65%, got us the classifier score of 0.84 or above. So the good news is that many times methylation gave us a definitive answer. The other way to look at it is that in about 1/3 of the cases, it didn't. So leaving room for, I think, ways we can improve the classifier. That said, what could we do with the data that we we obtained. So here you can see sort of how things were affected. About a third of the cases, we simply confirmed the diagnosis. Methylation gave the answer that was already suspected before methylation. Sometimes we were able to refine the diagnosis. I gave the example of medulloblastoma. We were able to subtype, or ependymoma. We was consistent. We found that it was an ependymoma, but we were able to show it's an ependymoma, REL-A fusion positive or YAP fusion positive, etc. Sometimes we were able to give a new diagnosis that wasn't suspected on initial review. Sometimes things were suggestive, but weren't definitive. And sometimes 10% of the cases or so, methylation simply was not contributory. We really couldn't improve the diagnosis based on the methylation class. And here's one way to look at the data. We're here on the left is the pre-classifier diagnosis. In the middle is the methylation class. And then on the right is the final integrated diagnosis. And there's some interesting things I think one can glean from the data. Down here are the ependymomas, things that were called ependymoma before methylation. And many times they mapped to ependymoma and were able to be subtyped to a particular class and mapped to ependymoma on final diagnosis. But sometimes was given a different diagnosis: medulloblastoma, glioblastoma, G34, MN1 altered glioma, etc. So the methylation had a contribution here, both refining the diagnosis of ependymoma, but also sometimes changing it. Here are pilocytic astrocytomas showing something similar, where many times it was confirmed, other times it was changed into an alternative diagnosis. And so this is sort of the impact. And you can see here on the upper right, when we track the initial diagnosis labeled here, pre-methylation, for example, astrocytoma IDH mutated, most of the times it was in green confirmed. So when it comes in as an IDH mutated astrocytoma, most of the time we confirm it. There's other tumor entities where it's less commonly confirmed. Pilocytic astrocytoma or anaplastic pilocytic astrocytoma, it was confirmed half the time, but other times we couldn't confirm it. Medulloblastoma, as I mentioned, methylation plays a big role in refining the diagnosis. And sometimes when the tumor came in as an unclassified entity, and that's shown here, it was given a descriptive diagnosis where methylation was able to many times resolve it. Not all the time. Sometimes we could only give it a descriptive diagnosis, but many times methylation could resolve it. And so that's sort of the impact of methylation in this particular case series. And I highlight GBM here because GBM is a diagnosis that's common, that most neuropathologists and pathologists can recognize. But even in glioblastoma, the most common intrinsic tumor we see, although most of the time methylation confirmed it, there were times where the methylation profile mapped to something else. For example, pilocytic astrocytoma, as I've shown you, pleomorphic xanthoastrocytoma, etc. Sometimes an IDH mutation was missed and it was mapped to an IDH mutant glioma. So even one of the more common scenarios we see of glioblastoma, occasionally methylation can have an impact on the diagnosis. And I've already shown you cases that came in as unclassified, that in many instances, we could come up by methylation with a specific ancillary diagnosis. And one thing we did when possible is when a case came to a specific class that had a mutational profile that was specific, or a copy number profile, et cetera. In many cases, we were able to then prove that. So for example, if it came in as a diffuse midline glioma K27M mutant, we were then able to show that K27M mutation, or the G34 mutation, or the FOXR2 alteration for this particular tumor type. So it wasn't that we used methylation only as the final arbiter, but when we could, we tried to supplement that with complementary data to come to a final diagnosis. And our experience is similar to what's out there in the literature. Here's a case, a paper from France looking at 62 pediatric tumors profiled over a couple of years in their network. And they showed qualitatively the same thing, that using the same 0.84 cutoff, that in their series, only 40% of the cases had that cutoff. So again, methylation is not perfect. It still requires some judgment, and it does not always give us the answer per se. That said, here's some of the impact as they saw it. That again, non-informative or inconsistent. Sometimes it gave a new diagnosis, novel proposition. Sometimes it was refined, and sometimes it was confirmed. And here's, I won't go into detail, but here's some of the breakdown on the individual tumor types. So methylation, I think, across other centers has an impact as well, although it's not the answer always in and of itself, and things need to be interpreted into context. It has technical, I think, challenges. One of them is tumor purity. Sometimes the tumors that pathologists receive are highly pure. Sometimes because of the specific tumor type or the tissue that's available, the tumor purity is low. Maybe it's on the edge of a lesion, or maybe that particular tumor type is just endowed with a large amount of non-neoplastic tissue. And so methylation has a challenge there, and sometimes does not give us the information we need when tumor purity is low and when the non-neoplastic elements are predominant. And we've found ways to address that here in our case series. We did find a number of tissues of tumors that we thought were probably neoplastic, but they had a lot of non-neoplastic elements and inflammatory elements, and they in fact matched to the inflammatory brain tissue. But we were able to informatically deconvolve this data and try to in silico remove some of that non-neoplastic element. We were able to resolve many of these tumors into a specific tumor type. And so we're just at the beginning of trying to resolve that. One example of how we can improve how we look at the methylation classifier. So this I've mentioned already, that on 1,000 tumors or so, this is the breakdown showing that methylation has an impact in many of these cases, but not all of these cases. And so there is work to do. There is experience, I think, is important, especially when the class score is low, and the ability to integrate that with other information and discussion with our clinical colleagues is really quite important. And so there are some surprises we've learned from methylation. One of them I'll just suggest here is that there are occasional cases that by histology look like one thing, but by methylation, they look like something else. And it's not to say they are that something else, but it becomes an interesting, I think, pattern for discovery on how we resolve when the histology doesn't match with what we expect it should match to by methylation. Here's a case here that pretty much any neuropathologist I know would call this glioblastoma, highly cellular, pseudopalliatic necrosis, etc. It was found to have a mutation that's not typical of glioblastoma, a BRAF-V600D mutation. And by methylation, it mapped to cases that were known to be pleomorphic xanthoastrocytoma. On the copy number profile, it had a PXA-like signature, which is loss of CDKN2A, and it did not have a GBM signature, which would be gain of 7 and loss, gain of chromosome 7 and loss of chromosome 10. Same here as well. We're finding that there are a number of tumors that mapped to the PXA methylation class that would not have been called PXA based on their initial histologic designation. And so that's where some discovery goes, and that raises the question, how will this patient behave? It looks like a GBM histologically, it maps to something else. And so further work, I think, needs to demonstrate how such cases, how these patients do. And I think it changes the way we think about how we define a particular tumor class. And it can be a humbling experience as well. Here's a case I had a couple of years ago in the lower lumbar spinal cord. The outside diagnosis was myxopapillary ependymoma. I saw the myxoid areas. I perhaps was lulled into a sense of complacency, gave it the same diagnosis. But we then ran methylation, and it turns out I was completely wrong. It was something else. It was a paraganglioma. Now, it doesn't look necessarily like a paraganglioma. I wouldn't have thought of it. At our consensus conference, we didn't think of it, but the data are the data. And so based on that, we then went back to this case, and we did follow-up IHC and showed that it was not positive for GFAP, which it should have been if it was a myxopapillary ependymoma, and it was positive for chromogranin marker of paraganglioma. So it can be a humbling experience where much of what we thought we knew by histology sometimes is not always correct. So our current practice is, when in doubt, we give a preliminary diagnosis, but then follow it up and wait for a definitive diagnosis until we can do the methylation. And here's some experience from another group from Denmark that published their experience on a number of cases. And I won't go over the detail, but the bottom line is most centers see about the same thing, where here they used a 0.9 cutoff, we used 0.84, but it's a similar kind of situation where in their series, again, about a third of the cases didn't match. And here they show the impact of the methylation classifier, and that a number of these tumors don't match. So there is work to do. So what about these cases that don't match? There's a number of reasons for that. I mentioned technical issues, DNA quality. The case could be an old case, so that can affect the methylation process. Tumor purity can be an issue. The other reason is that the tumor in question is not represented in the classifier. So the classifier is a reference set, and you compare that tumor to the reference set. If the reference set doesn't have that entity, then methylation is not going to give you the answer. And so it could be a new entity. And that's the discovery concept here. That's how the classifier can grow over time with accumulation of more data into a central repository. And here's one example here of how that could be leveraged. We noticed in the process of doing these cases that we found a number of tumors that were close to pleomorphic xanthorhabdocytoma, but we're not in the cluster. The classifier gave a low score for PXA or something else. And on this UMAP, they were near PXA, but they weren't in it. And so they really weren't in the classifier. And then we started looking at other characteristics of these tumors. Methylation does provide copy number aberrations as a side benefit. So we could see that virtually all of these tumors had loss of chromosome 13 and also sometimes chromosome 17. We found that many of them had NF1 mutations. Many of them had other kinds of mutations. And furthermore, they didn't have PXA features. They didn't have that CDKN2A homozygous loss that we like to see for PXA. So what are these? They weren't in the classifier. They seem to be clustering together. They seem to have other uniform characteristics. Could they be something else? And how could we unify this? Here's some of the histology of these cases that map to that cluster. You can see a great variety of histopathologic appearance. Sometimes the cells had perinuclear halos, sort of like an oligodendroglioma. Sometimes they had palisading of nuclei, like an ependymoma. Here's more ependymoma-like structures. Sometimes they look papillary. Sometimes they looked extremely pleomorphic, like a PXA. For example, sometimes they looked something different. And so one can imagine why these tumors had not been classified before, because they didn't have histology to unify them. But now with methylation, we can try on a biological basis to unify them. And we gave it an initial name of this, which could be changed in the future, but this was our best name, high-grade glioma with pleomorphic and pseudopapillary features. We were able to show that it's distinct from other entities and clusters separately from them, et cetera. And again, when we compare it to PXA, it has that chromosome 13 loss. It does not have CDKN2A loss, and compared to another entity, same thing. So it has other features that distinguish it. And there's some clinical implications here as well. If we look at the age of patients with this provisional entity of HPAP compared to glioblastomas, PXAs, pilocytics, et cetera, it had a distinct age group. Young adults, around 50 was the median age or so. And then, of course, we need to know, why does this matter? Is there a clinical implications here? Some of these cases did come in with a glioblastoma histopathology diagnosis, but here we can see that these HPAPs patients, again, small sample size, only 30 or so patients, but seem to have better outcome than patients with glioblastoma, and also better outcome than patients with pleomorphic xanthoastrocytoma. So there could potentially be some clinical implications to getting this right as a diagnosis. And so just one other example that we're working on now, and that is a subtype potentially of posterior fossa ependymoma. And so many of you know that ependymoma, although in the past was given sort of a single diagnosis of ependymoma or anaplastic ependymoma, now is subdivided according to its location in the three major compartments: supratentorial, posterior fossa, and spinal cord. And it's been known for a number of years now that some molecular alterations can define these. For example, Relay fusions are pretty much specific to supratentorial tumors. They don't generally occur in tumors of other compartments. Posterior fossa tumors can be divided into subependymoma, ependymoma group A and group B, et cetera, et cetera. And so it was found reassuringly that these classes also have their corresponding methylation defined group. They can be separated from each other based on methylation. And so that's sort of the current thinking on ependymoma, and that's pretty much represented in the WHO with some recent additions. But what we found was a rare case that it was in the posterior fossa, and it looked by methylation like a PFB, posterior fossa type B, but it wasn't quite there. The score was low, didn't cluster quite with it, and it was sort of outside that group. And by chance, that tumor had been sequenced, and a mutation in ACVR1 that had not yet been described as ependymomas was noted. So we sort of filed that away. Isn't that interesting? And then over time, additional cases collected that by methylation mapped to this same case, the same index case, and we sequenced those, and we also found ACVR1 mutations in those. Again, not getting to this concept of a new entity, and we can show some of the results here. Here we've got PFB, which now can be subdivided into subclasses, but these are all PFBs. We have posterior fossa type A ependymomas. Here's some other ependymoma-related entities as well. Here are the ACVR1. We found them to be trimethyl positive as compared to PFAs, which are trimethyl negative, but they were a distinct entity. And what's the clinical implication? You can say, well, so what? But potentially there could be some clinical relevance to that, and that's because diffuse midline gliomas are known to have ACVR1 activating mutations, and there is interest in development of clinical trials that target this ACVR1 activating mutation in diffuse midline glioma. And so here's some of the other features of this tumor type. Here you can see the group that all have ACVR1 mutations, and they're distinct from diffuse midline gliomas that are ACVR1 mutant based on their mutational profile. The specific mutations that occur in the ependymomas seem to be similar to the ones that occur in the diffuse midline glioma. These are hotspot mutations that overlap between the two tumor types. So these are activating oncogenic mutations. And it's interesting to us that looking at these ACVR1 mutant tumors, there's at least preclinical data to suggest that clinical trial could be considered for to target ACVR1 in diffuse midline gliomas. And so that raises the possibility that these tumors in the future could also be targeted. And so what kind of lessons are we learning from using methylation in brain tumor diagnostics? And that is the classifier in methylation can raise a hypothesis that wasn't previously entertained, that wasn't thought of. But then that could be a hypothesis that could then be tested and confirmed or refuted. I mentioned examples of H3 mutated gliomas, either K27M or G34R. Mutated gliomas can then, if suggested by methylation, can then be confirmed by either immunohistochemistry or sequencing. Another, I think, application is that there's some entities that really, I think, are emerging that can only be confidently diagnosed by the methylation classifier. That includes many pediatric embryonal tumors. We can get to an embryonal neoplasm by conventional means, but we can't get more specific, and methylation can help classify that. There's other entities, I list this one here, that realistically can only be, the histology is not specific and can really be only diagnosed by methylation. That said, although helpful in these situations, I think, as I've shown you, there are cases remain. The classifier is not perfect, and there are a number of cases that simply cannot be classified by methylation for a variety of reasons, technical, maybe there are rare cases or what have you. So there's more to learn. New entities likely exist, and the classifier is only as good as a reference set on which that tumor is compared with. And so the larger and more representative the reference set, the better the classifier and the better we're able to accurately diagnose cases. And again, raising the advantage of combining data rather than siloing that data. So just in my last few slides, I think I've probably shown this already, but basically the methylation process starts with FFPE tissues. Fresh frozen can be used as well, but typically we use FFPE tissues and undergoes the hybridization process, etc. We then use a classifier and machine learning and clustering techniques to help us get to an entity. We then compare that to orthogonal data, copy number data. We can detect 1p19q co-deletion for oligodendroglioma, EGFR amplification, etc. That helps us confirm or refute a particular diagnosis. We can identify MGMT promoter methylation, etc. So it's really the integration of these all things together that gets us to an integrated diagnosis. And I showed this slide at the beginning that really it's an integrated approach using all the relevant data that's available and ensuring that everything can fit. And here, methylation doesn't replace any of these modalities, but it can add to it, particularly with difficult to diagnose cases. So I'll summarize by hoping that I convinced you that methylation profiling has some practical advantages as an additional tool, not to replace what we have, but to add to it. And with experience, I think it can improve the accuracy of diagnosis for particular cases. It has advantages of being technically feasible, amenable to FFPE samples, and it's relatively stable over time and across tumor regions. It by no means solves all of our problems, and it needs to be interpreted in context. One cannot ignore the other information because methylation is just one part of the armamentarium of tumor diagnostics. And that additional information is absolutely critical to integrate with the methylation. And sometimes we need that. Sometimes we need the genomic information that could help us get to a specific diagnosis or a specific targeted therapy for that patient that methylation can't provide. So more can be done to learn how to utilize this and integrate this into classification into daily practice. So with that, I will conclude and thank you for your attention. I'd be happy to discuss or answer any questions.
Thank you. Thank you very much, Dr. Aldape. This is a fantastic lecture. It's certainly given me a lot of insight into methylation and it's the science behind it, as well as its application and its limitation. And so this is very useful, and all these lectures are recorded, and I'll speak to the speakers whether they can be on public domain, but all of you can review it. So thank you very much, Dr. Aldape. Dr. Mille, do you have any questions?
I don't have any questions. I guess I also want to thank Dr. Aldape very much for his talk, and also for his ability to do this on short notice, since we've all put this course together a very little lead time. So very much appreciated, Ken.
Thanks for the opportunity.
Thank you. Can I just ask Dr. Cohen, if you are there, do you have any questions, comments? Dr. Aldape, I have got a few practical questions to ask. This is because as a neurosurgeon, I don't. Dr. Cohen is here. Dr. Cohen, do you have any comments, questions?
Can you hear me?
Yes, we can. Yes, Dr.
I'm
Cohen.
sorry, I'm calling in from the OR from Johns Hopkins, but I just wanted to say that was a beautiful talk. And again, I think the future is going to be in this hybrid diagnosis that's coming out from CNS5, and the ability to use this information to help therapy. But no, it was a beautiful talk. Thanks so much.
Thank you.
Thank you. Dr. Aldape, in terms of the amount of time for you to give the diagnosis, and in terms of the expense, and Dr. Mille previously mentioned the reference set, if I got it correct, that the reference set is mainly based in Germany, and can you comment on those three questions in terms of expense, time, and the reference set? Thanks.
Yeah. Time, practically speaking, it's about 2 weeks. It takes us a few days to isolate the DNA, and we have to do by-cell-by conversion, and then we run it, etc. Sort of from soup to nuts. We're at about 2 weeks turnaround time. The costs on the reagents, the juice that you need to do it, it's about $300 per case. Then there's the labor, etc. But about $300 in disposables for costs. And the reference set, you're right. The initial reference set was based in Germany. We are developing our own. We would like to make it publicly available. If I can share my screen real quick. What we'd like to do is we're developing this portal that would be available to the public, to registered users. To date, we've got about almost 14,000 CNS tumors, and this is something where someone could run methylation either through us or through their own lab, and they could upload their data onto this portal, and then they can interact with this data. They can look to see if their tumor matches to ependymomas PFA. They can see where it goes. They can potentially look at characteristics of what's known about these patients. We can show here's the Kaplan-Meier survival of this. We can compare that.
We can't see you. We can't see your screen, Dr. Aldape.
Oh, I'm so sorry.
Sorry.
I'm so sorry.
So that's okay.
Okay, so I apologize. So here is the portal that I'm talking about that shows about 14,000 brain tumors that we've collected to date. One can upload their sample in this and then see where it maps. For example, if it goes to ependymoma posterior fossa type A, one can then interact with the data. And if there is survival data on these patients, we can look at that, can compare it to patient survival from, for example, this is posterior fossa ependymoma type B, et cetera, and do these kinds of things. So this is sort of one of the efforts we're trying to do to democratize this. and to take advantage of the fact that the classifier depends on a large and diverse reference set. And then the tumor that's uploaded by individuals can then be used to add and further develop the classifier. So these are some of the ways that we're trying to move this forward. Also, I think it's going to promote standardization across centers. One wants to have similar diagnostics and criteria so that a methylation class at one institution gives us the same answer as a methylation class across another.
That's great. Thank you. Thank you very much, Dr. Aldape. I'm just going to give the chair back to Dr. Mille. Thank you, Dr. Mille.
Thank you. Is Dr. Hawkins on?
Yes,
Yep.
Dr. Hawkins on. Yeah.
Very good. Then I think what we will, looking at the clock, we have 1 minute to 10, so I think I will start introducing Dr. Hawkins and give her the maximum time she needs to talk. So Cynthia Hawkins is a MD, PhD. She's a molecular neuropathologist. Got her MD and PhD degrees from Western University in Canada, and did her residency in neuropathology at the University of Toronto. During her residency, as I understand it, she took time to go to Zurich, University of Zurich, and did a postdoctoral fellowship there. She has been at the Hospital for Sick Children as an attending pathologist since 2002, and she's currently medical director of the Translational Molecular Pathology Laboratory and group. She's a senior scientist of the Sick Kids Research Institute, and she's a professor of laboratory medicine and pathobiology at the University of Toronto. I want to particularly thank her, not just with all the speakers of doing this on short notice, but of being very flexible with her schedule so that we could work around some last minute changes we had to make. So thank you, Cynthia, very much, and I will let you take the podium.
Great. Thanks so much for the invitation. I will try doing the screen share here. All right, you guys should be able to see now. Is that correct? Okay, so I'm going to cover pediatric gliomas, both low and high grade. I'm going to try to give you an approach to how we diagnose these tumors now using a combination of morphologic and molecular techniques. Show you the spectrum and relative frequency of these molecular alterations in pediatric gliomas, and then hopefully give you an idea of some of the differences between pediatric type and adult type low-grade gliomas, and why that's important for patient management. So just as a bit of context, back in, well, it was before 2016, but introduced in the WHO in 2016, sort of that the goal of that revision of the WHO classification was to try to sort of bring in or introduce this concept of how we could structure the diagnoses of CNS tumors in the molecular era. And that sort of sprung out of some work that we had done earlier in the Harlem consensus meeting, this concept of an integrated diagnosis where we try to incorporate both the molecular and the morphologic information into a sort of overarching diagnosis. And that molecular data can include sequencing, it can include copy number things, it can include methylation, basically, or even immunohistochemistry. So any of these ancillary things we do beyond looking at the morphologic characteristics. And a number of molecularly defined entities were introduced back in 2016. And this that trend continued in the most recent 5th edition in 2021. There were some other major changes that happened in 2021. So at least for the adult type gliomas, there was an attempt to sort of move away from assigning grades based on a name to grading within an entity. And so, whereas we used to, for example, call a grade 4 IDH mutant astrocytoma, glioblastoma, that's not the case anymore in the new classification. And that's to try to align a little bit more with what happens in non-CNS tumors, where people will grade within a specific tumor entity. There was a change from the Roman to Arabic numerals. So you may see that in reports that you get where, the grade is now given a Roman numeral, an Arabic numeral. And particularly, there's been a lot of changes, but I'm going to specifically talk here about the diffuse gliomas. And I think one of the huge things that we were able to incorporate was this distinction between pediatric type and adult type entities. And of course, addition of more molecularly defined tumor types. So this is maybe small depending on how big your screen is, but it gives you a sort of an overview of what the classification of gliomas looks like now. And I've just highlighted in red these pediatric type entities that have been added, and these adult type ones have been more specifically delineated. There's a number of other molecularly defined entities that have been added, which I've also highlighted in red here. So just if we think about this conceptually, if you see a diffuse glioma, you can kind of think about it in terms of those that generally arise in the adult age groups. And I should stress that, adult type and pediatric type, that doesn't mean that adults can't get pediatric type cancers and vice versa. It just means that they're much more enriched in that age group. And this is particularly important to think about in the adolescent young adult or AYA population, where they really do get a mixture of adult type and pediatric type cancers. And it's important to remember that if you're dealing with a 25-year-old or a 30-year-old with a diffuse glioma, they very well may have a pediatric type glioma. And the clinical consequences of that are quite a lot. So I'm not going to spend a lot of time on the adults type stuff, but I'll just give you some of the highlights of how that's changed. So we still have astrocytoma IDH mutant. Those can be from grades 2 to 4. Grade 3 means you have an elevated mitotic index as well as anaplasia. Grade 4 means you found microvascular proliferation, pseudopalissating necrosis, or importantly, you found a homozygous CDKN2A deletion. So if you have something that looks like grade 2 or grade 3 astrocytoma with an IDH mutation, you should check for CDKN2A homozygous deletion, because that will automatically bump it to grade 4 in the new WHO. Similarly, for oligodendroglioma, again, you still have to show it's IDH mutant. You have to show it has 1p19q co-deletion. These can be grades 2 or 3, with grade 3 being those with microvascular proliferation or necrosis, or homozygous CDKN2A deletion. So again, for grade 2 oligos, the CDKN2A deletion becomes important. And then there's the glioblastoma IDH wild type. These are all grade 4. Many of these can be diagnosed based on morphologic features alone. So, our standard microvascular proliferation, necrosis. There are also molecular features that can give you a diagnosis of GBM, even if you don't have all the morphologic features. In particular, EGFR amplification, gain of 7, loss of 10 in terms of copy number changes. TERT promoter mutation is also included in this. We were actually just talking about this yesterday in Ken's meeting, where we talk about the methylation data. So this is technically a feature that would give you enough to call it a glioblastoma. However, it should be a little bit careful, because that's a little more promiscuous than perhaps was thought in the original in those original molecular diagnostic terms. And so if that's the only thing you have and nothing else smells like a GBM, I would be careful using that criterion. Right. And so that in particular, younger patients, the morphology doesn't really fit. And I give the example there, BRAF Kennedy mutant gliomas, diffuse gliomas can have gain of 7, loss of 10. Those are not GBMs, and they do not behave like GBMs. And the other thing, and I'll bring this up a little bit later as well, but it's really important to consider if you have high-grade gliomas in this adolescent young adult population, consider the possibility of replication repair deficiency, and a mismatch repair, even in IDH mutant gliomas. So many of them will actually have IDH mutant gliomas as well. And I'll show you some features of that later. So this is just sort of an approach you can use for the adult side of things. So obviously, if you get a diffuse glioma, the most important thing to look for is IDH. The R132H can be done with immunohistochemistry. And then you break it down into those that have P53 and HRX. Again, that can be done with immunohistochemistry. And if those look morphologically like a grade 2 or a grade 3, then consider doing the CDKN2A. Those are negative, then you would be looking for 1P19Q. Again, the recommendation is to try to use a technique that will find the whole arm loss. But, if all you have is FISH, that's also, can be used. It's just worth remembering that there's caveats there. And then again, if it looks like a grade 2, you consider doing the CDKN2A, and that can also be done with FISH. If the patient's less than, and some people say 55, and you do not have the R132H, then you should consider looking for other IDH mutations, the less frequent ones. And then again, if you find one of those, you're back over here. If you do not find any IDH mutation, you're most likely dealing with an adult type glioblastoma. Maybe in the AYA population, consider pediatric type gliomas, but most of these are going to fall into this category. Okay, so I'm going to go through a few cases, and then we'll talk about some of the differentials and approaches for the pediatric type gliomas. So this was a 14-year-old girl, and she presented with seizures and a headache, and she had this large, sort of minimally enhancing infiltrative mass. So this is what it looked like morphologically. So pretty classic high-grade glioma look. So areas of necrosis with a little bit of pseudopalisading, quite anaplastic looking, more multinucleated cells, sort of typical high-grade glioma. Immunohistochemistry showed glial differentiation, so GFAP positive. Diffusely P53 positive on immunohistochemistry, so that generally implies that there's an underlying P53 mutation, and there's loss of ATRX. So again, suggesting there's an ATRX mutation. So what do we know about the pediatric age group in terms of high-grade gliomas, and what should we be thinking about? So overall, in contrast to adults, in pediatrics, the high grades are actually less common than the low grades. They're usually not the result of a progression from a low-grade counterpart. So unlike the IDH mutant gliomas, which can sort of inevitably progress to high grades, that's much less common in pediatrics other than the BRAF V600Es, which can do that. And again, these are molecularly distinct. So by definition, these are IDH wild type. So in contrast to the adults, the most common mutations that you see in high-grade gliomas in the pediatric age group involve the histone genes. We don't really have good population-based data, but they're probably about half of the high-grade gliomas in children, particularly if you include the brainstem gliomas, which aren't always biopsy. So we get two major types of histone mutations in pediatrics, one involving the lysine 27 on histone 3. These are largely in the midline. And then a less common one in hemispheric gliomas involving the glycine 34. So these are two different subtypes of gliomas, high-grade gliomas in the new classification. So there's the diffuse midline glioma involving H3K27 and the diffuse hemispheric gliomas involving H3G34. So in terms of diffuse midline gliomas, used to be called H3K27M mutant. In the new classification, you'll see it's H3K27 altered, and I'll go through some of the reasons for that slight change. So these K27 altered cases are enriched in childhood, but importantly, they really can occur at any age. This is a nice paper from David Solomon, UCSF group, that showed, that you can certainly get K27M mutations up into your 60s or even 70s, particularly once you move outside the pons. So a lot of the thalamic gliomas or spinal cord gliomas can occur that are K27M mutant or K27 altered can occur in older patients. A difference, I guess, sort of subtle difference between the G34 and the K27 is that the K27 can occur in this H3.1 protein as well as the 3.3. So depending on how you're looking for that, you can miss these. The antibodies should pick up both, although sometimes it's a little less sensitive for the 3.1 mutations. And of course, if you're using sequencing, then you would need to make sure that your panel is covering both of these genes. Overall, these have a really quite poor outcome. This is just data, again, from Chris Jones' Cancer Cell paper. The green line are the K27M mutants. So compared to pretty much, high-grade gliomas in the pediatric age group, these are pretty much the worst. Morphologically, these ones can be, they're always, pretty much always glial. I hate saying always, but these are like 95% glial. About 2/3 of them are going to look classically like GBMs, but it's important to remember that they can look more like what we traditionally would have called anaplastic astros, so kind of just some mitotic figures. And then there's about 10 to 15% that actually will just look like what we would have called a diffuse astrocytoma. So there's no mitoses, it's just kind of hypercellular, diffusely infiltrative. They can have really low MIB1. And that can lead to some sort of confusion in the diagnosis and what does this really mean? These, about 60% of them will have a P53 mutation, and about, depending on your age. So if you're younger, low chance of ATRX. If you're older, it can be up to 50% of them will have ATRX. But this is much less common than with the G34 mutant tumors. Just an important thing to remember. So the current recommendation for using the term diffuse midline glioma H3K27 mutant or H3K27 altered is that you have an actual diffuse glioma. So if you see something that looks non-diffuse, like circumscribed pilocytic ganglioglioma, then you should not be using this term. However, it is important to remember that even in these non-diffuse gliomas, this is a very poor prognostic marker. So there's a nice study. Drew was the first author here. This is when he was in Michigan. And we have similar data. So even if it looks like a low-grade glioma morphologically, the patients that have K27M are going to do more poorly than the patients who do not. They will pretty much all eventually die, although they may live, up to a decade, rarely. Just a little bit about the mechanism so that you understand what the immunohistochemistry is doing and why it can be helpful for picking these mutations up or this whole group up. So the K27M mutation in histone 3 inhibits the PRC2 complex. So PRC2 is a sort of complex of proteins. The active component, the methyltransferase component, is called EZH2. And that is responsible for depositing this trimethyl mark on lysine 27. So obviously, if you change the lysine to a methionine, you cannot put a methyl group on there. But importantly, this actually inhibits this whole complex. So it means that even the wild type histones don't get methylated. And so if you use an antibody for this H3K27 trimethyl mark, most normal cells will have brown staining. So most cells have enough of this in their nucleus that it'll look brown. And so if you look, on the slide, you'll see all the normal endothelial cells and so on will have brown staining in their nucleus. The tumor cells have all lost this, and that's because of that dominant negative effect of having the methionine there. You can also use an H3K27M specific antibody that should also show diffuse nuclear positivity in all of the tumor cells. This antibody can be a bit tricky. It's not particularly robust. So if you have a case where there's been cautery or the block has been sitting for a long time, something like that, autopsy material, for example, this antibody will not work. The K27 trimethyl antibody is much more robust. Less specific, but more robust. So, as people started doing that antibody, using the K27M antibody and the K27 trimethyl antibody, they found that some diffuse midline gliomas had loss of the K27 trimethylation, but did not have a K27M mutation. And this is a similar pattern actually to was seen in posterior fossa type A ependymomas. And that was a nice paper from Sriram Vanetti. And then David Ellison's work showed the mechanism of that. So we sort of wondered for a long time, why are these PFA type ependymomas losing the K27 trimethylation? Because we knew that it was exceptional for them to have the K27M mutation. And it was found that there was another protein that was overexpressed in both posterior facet type A ependymomas, as well as in some of these diffuse midline gliomas, originally called CXOR67, now called EasyHip. And so that sort of identified a quite rare but definite subgroup of diffuse midline gliomas. This can occur in the thalamus, it can occur in the brainstem, where they have a loss of K27 trimethylation, no K27M mutation, but they do have overexpression of EasyHip. There is an antibody also to EasyHip. It's actually quite a good one. And so using that in combination with the K27 trimethyl antibody gives you a nice pattern where you see nice positivity for EasyHip and loss of the K27 trimethylation. And that allows you to find these diffuse midline gliomas as well as the PFA type ependymomas. When people looked at the outcome of patients that had this H3 wild type with overexpression of EasyHip, they saw that the age range and the outcome was very similar to patients that actually had a mutation in K27M. And so it was felt that this is representing, again, a rare, but another sort of variant of these diffuse midline gliomas. And then the final group that's been added are the bithalamics. So everyone, every clinician is very familiar with this classic appearance, bithalamic glioma. And, it was quite surprising actually when we first started to look at these and realized that they only very rarely actually have a K27M mutation. And then again, this is a nice paper from the UCSF group, David Solomon, showing that many of these will have instead this EGFR exon 20 insertion. Many, but not all, will also have the loss of K27 trimethylation. And so there was a bit of debate, but in the end, these went into the same group with the other K27 altered diffuse midline gliomas. So if we think about the options now for these midline gliomas, you can have diffuse midline glioma with the H3.3 K27M. You can have it in one of the other histone genes. The 3.2 is exceptional. And then you can have the ones that are associated with EGFR mutation. These are mostly bithalamic gliomas. And then this type where there's no actual K27M mutation, but you have loss of trimethylation because of this EasyHip overexpression. Once we move out of the midline, then we're dealing with a sort of spectrum of hemispheric gliomas. Again, you can sort of broadly think about these based on the age range in which they most frequently occur. Again, it's not exclusive to these, but this is where they're most frequent. So the adolescent young adult population most commonly will get the G34 mutation. And then you have in pediatric, in sort of the childhood age, you have the diffuse high-grade glioma that are H3 wild type. And then there's the infantile types. So if we look at these G34 mutant diffuse hemispheric gliomas, again, this is really an adolescent young adult disease. So this is curves showing the sort of population spectrum or age range of when we most frequently see these alterations. The green is IDH, and then the red is G34. So they come on a little bit earlier than the IDH on average, but they're peaking in the 20s to 30s, and then they really tail off. But it really is a differential with the IDH mutant gliomas in patients that are in that adolescent young adult population. These are much rarer than K27M. So in our population-based cohort in Toronto, these G34 mutants account for maybe 5% of the hemispheric gliomas. So rare, but definitely happens. They are highly associated with mutations in TP53 and ATRX. So over 90% of the cases will have mutations in these two genes. About 40 to 50% will have also a mutation in PDGFR alpha. May be important if you want to be able to offer these patients a targeted therapeutic, for example. Their outcome is generally bad, probably a little better than the K27s, but K27Ms, but still bad. In contrast to the K27M mutant gliomas, these are usually MGMT methylated, although the response to temozolomide is not what it is in IDH mutant gliomas. And if we just look at outcome, again, this is a little faint and small, but the red line is the G34 mutants. The blue line is the IDHs. So they're much worse than IDH mutant gliomas. Morphologically, most commonly these will be glial, but unlike the K27Ms, they do have a propensity to have a more neural phenotype sometimes, including stuff that looks like what we used to call peanuts or sort of an embryonal look. And so it's an important thing to just keep in mind. Sometimes, consult cases from patients in their 20s where someone thinks it looks like a peanut. They're almost always G34 mutant. We call them gliomas. They can even get, actual ganglion cell differentiation rarely. This is the classic immunoprofile. So diffuse P53 positivity, ATRX loss, so very similar to the IDH mutant astrocytomas. Some will have a lot of GFAP, but not all. So it does not have to have GFAP. They almost all will lose expression of OLIG2, which is pretty distinct for a glioma. Most gliomas will have OLIG2 retained. And there is an antibody for the G34R. There's also one for the G34V, although that is a very rare mutation. And that works pretty well to pick up these cases. The main thing is just to consider this if you have an IDH wild type high-grade glioma that has P53 positivity and ATRX loss. The other option you could have other than a non-canonical IDH is G34. Okay, so what about the other type, diffuse high-grade pediatric type gliomas that don't have a histone mutation? What else can they have? So some will have BRAF V600E. MYC-N amplification is another option. Some will have separate tyrosine kinase mutations or amplifications, and then importantly, some will be mismatch repair associated. And the more we start looking for this, and I don't have like accurate prevalence data, we're trying to come up with this, but this is actually much more common than you would think, and also depending on the population that you serve. But certainly in Toronto, where we have an extremely multicultural population, this is not uncommon in the adolescent young adult age group. Some important points for these different types. So think mistakes I've seen. So BRAF V600E positive high-grade gliomas or glioblastomas, they can be associated or usually associated with CDKN2A deletions. You can see K27M as a co-mutation. They can have HRX mutations and they can have P53 mutations. Importantly, for a pathologist, they can look really epithelioid, and so they mimic other tumors. If they're in the brain, I've seen them called ATRTs. They can even actually have, a lot of loss of SMARCB1. So if you see SMARCB1 loss in the context of a BRAF V600E mutation, that's not an ATRT. That is a BRAF V600E mutant high-grade glioma. The other mistake I've seen is them being called rhabdoid meningiomas. So these will crawl out into the leptomeninges quite frequently, and because they're so epithelioid looking, they sometimes look meningothelial. And again, BRAF V600E mutant meningioma would be a very weird diagnosis. So it's just something to consider, especially in the young adults. Okay, mismatch repair deficiency. So everyone is probably familiar with Lynch syndrome. So Lynch syndrome means you have one mutation in one of these mismatch repair genes. It's most frequently MSH2 for Lynch. Classically, these patients will get GI or GU cancers in their 40s or 50s. But we do see gliomas arising in the adolescent young adult population in Lynch families. So it is an important thing to think about. The other thing we see, and this is really a pediatric thing, is biallelic mismatch repair deficiency. So those are patients that have 2 hits in one of the mismatch repair genes. It's most frequently PMS2, but can be in any of them. So if you serve a population where there's a lot of consanguinity, it's important to consider this BMMRD or CMMRD. The mean age of diagnosis here is 9 years, and many of these will present with high-grade gliomas. So it is something to consider. So when to think about it. So the CMMRD patients, the congenital biallelic patients, often will have almost an NF1 look. So they can have multiple cafe au lait spots. They may even fulfill criteria for NF1. They usually, though, have normal growth and a normal IQ, and obviously will not have NF1 germline mutations. There's often a family history of GI cancers and a family history of consanguinity. However, it's important to remember that the Lynch patients that are coming in as teenagers or in their 20s often will have no family history. Sometimes if you dig, you'll find somebody that had a colon cancer, but not always. And so, you might not be able to rely on this. They also will not have any of these other, skin manifestations or things like this. So it's just important to think about it. From a pathology perspective, and this is not universal, but it's a clue. So if you see an extremely pleomorphic looking glioma with lots of large multinucleated cells, severe anaplasia, you might be even thinking, is this an anaplastic PXA, or is this a giant cell GBM? Those are the patients you really should think about doing the mismatch repair protein immunohistochemistry. Now, the important thing to remember as well here is that these mismatch repair deficient gliomas will have alterations that you see in non-mismatch repair deficient gliomas. So just because you find an IDH or you find a P53, that certainly does not exclude mismatch repair deficiency. And in fact, almost all of the mismatch repair deficient cancers will have diffuse P53 immunopositivity. So, that would almost be a common pattern. IDH is quite frequent as well. I would say probably 20 to 30% of them will have an IDH mutation. The pattern you're looking for in terms of the immunohistochemistry, and again, if you're familiar with Lynch, then the biallelic ones can be confusing. So normally in a cancer that is not mismatch repair deficient, you should see nuclear positivity for these genes in every cell, normal cells and tumor cells. So you would see this type of pattern if it was normal. In the biallelic patients, you will actually lose that immunostaining in every cell, normal cells and tumor cells. And so while you might think that your immunohistochemistry didn't work, this is actually the pattern you're expecting to see. That's in contrast to the Lynch patients, where the non-mutant genes will all be retained and the mutant gene will be retained in the normal cells and lost in the tumor cells. So those are the two different patterns that you can see in these mismatch repair deficient patients. Okay, so sort of a potential approach to seeing a high-grade glioma in a pediatric age group. First is to consider, is this in the midline or is this in the hemispheres? If it's in the midline, then you're thinking about the K27 altered cases. So maybe do the K27 trimethyl and the K27M. And then you can do other immunos to sort of further subclassify those. If it's in the hemispheres, and if it's a child or an adolescent or a young adult, then consider the G34. If it's an infant, then you would think about these other tumors, and I'll talk about the infants afterwards. And in the particular case that we're talking about here, we saw these giant cells, sort of severe atypia. We did the mismatch repair proteins. This patient had complete loss of MSH2. So this was a patient with a congenital mismatch repair deficiency. We then went on and did mutation burden testing. So this patient was hypermutant, had more than 10 mutations per megabase. And this patient got immune checkpoint blockade and actually did quite well with that treatment. And we've had some long-term survivors of high-grade gliomas that had mismatch repair deficiency, hypermutation, and were given immune checkpoint blockade. So it is particularly important, not only for the families in terms of screening for their mismatch repair deficiency, but for these patients, because they can get long-term survival if they get access to immune checkpoint inhibitors. So some of the key take-home points then for the diffuse high-grade pediatric type gliomas. So the G34s, these are hemispheric tumors. They're enriched in the adolescent young adult population. They have a poor prognosis, but it's a bit better than K27M with a median survival of about 18 months. They can look like high-grade gliomas, or they can have this peanut-like histology. So it's important to think about that if you see that morphology. K27M is midline. This is enriched in children, but it can occur at any age. They have a very poor prognosis, particularly if it's a diffuse high-grade histology with a median survival of only 10 to 12 months. They can have a broad range of histologies. All of the diffuse astrocytomas, also pilocytics, ganglios, even ependymomas can have this mutation. With these other morphologies, circumscribed morphologies, they're still generally fatal, but there is a chance of a longer survival if you have a non-high-grade histology. Important to remember that high-grade gliomas in the pediatric age group can have more than one driver. So, and some of these are really important in terms of guiding therapy. So obviously the BRAF V600Es can get targeted inhibitors. The mismatch repair deficient can get immunotherapy. If they have a receptor tyrosine kinase, you can sometimes access targeted inhibitors for those patients. And then, of course, important to think about it, if it doesn't have any of these things, is it really a pediatric type high-grade glioma, or are you dealing with an adult type IDH mutant glioma? Okay, the next case is an infant, a 4-day-old, presented with restlessness, and then on imaging had this huge intracranial mass with an intratumoral bleed. And so that patient had a surgical debulking, and it looked like a high-grade glioma, very cellular, lots of mitoses, some areas of pseudopalisade necrosis, diffusely GFAP positive. So what do we know about infant type gliomas? So CNS tumors are actually the most common tumors that occur in children under the age of 1, and about 40% of those are gliomas. They have actually paradoxical outcomes. So low-grade gliomas in infants do worse than you might expect based on older children, while the high-grade gliomas actually do better. Overall, they're kind of underreported in the medical literature, and prior to the last few years, there was really only anecdotal data regarding their molecular features. We did this big multinational study trying to look at what are the genetics underlying the pediatric or the infant type gliomas. This was led by one of our neuro-oncology fellows who's now in Zurich, Anna Strucklin. So what we found looking at the molecular of the infants, and this included both low and high grade, is that there were 3 main subgroups. So in the midline, most of these are low grade, and most of these are driven by RAS-MAP kinase alterations, including BRAF fusions and BRAF V600Es. And then in the hemispheres, they kind of split out into those that were RAS-MAP kinase driven, and those that were driven by other alterations, in particular receptor tyrosine kinase fusions. And these are important to recognize because these fusions, which can occur in ALK, ROS, NTRK, or MET, are potentially therapeutically targetable. And these patients actually do quite a bit better than you might expect. And so this has been included now in the WHO as a sort of distinct category of infant type hemispheric gliomas. And so if we look at, these are largely high-grade gliomas. This red line are the hemispheric receptor tyrosine kinase fused. This is the survival for the hemispherics that are RAS-MAP kinase driven and are low-grade. But, you're still talking over 50% long-term survival for high-grade glioma in an infant that had one of these fusions. And so it's important from a prognostic perspective, and it's important because you can access targeted therapeutics for those patients. So important thing to remember. So in terms of the algorithm, then, if we're looking at hemispherics and we're in an infant, if you can, it's really nice to be able to try to look for these fusions. So in this particular patient, we did an RNA-seq based NGS platform and found this PP1CB ALK fusion. This patient was treated then with, had a subtotal resection actually, was treated with chemotherapy and an ALK inhibitor. And this patient is still alive. I think it's actually been more like 5 years now with stable disease. So it's important to think about this. It's important to remember that many of these patients actually are dying because of the size of the tumor and getting hemorrhages because of the big surgery. And they may actually respond to some of these targeted therapeutics, so may not need quite such an extensive surgical approach. They also, interestingly, and this is sort of more anecdotal, but we have a few cases where the patients came back 3 or 4 years later and had the rest of their tumor removed when they were a little bit older, and they can mature over time. So it's, I think, a very different biology than what we're seeing in terms of high-grade gliomas in older children or in adults. So some key take-home points then, if you're looking at a glioma in the infant population, definitely think of these as very distinct. If they have midline RAS-MAP kinase tumor, they're largely low-grade glioma, and they actually have a worse outcome than the older patients, where the outcome with these RAS-MAP kinase-driven ones is quite good. In the hemispheres, if they look low-grade, again, they're usually RAS-MAP kinase. These ones have a really excellent outcome, so long-term survival is close to 100% for those patients. If they have something that looks more like a high-grade glioma, then consider these receptor tyrosine kinase fusions, NTRK, ROS, ALK, MET fusions. And these may respond well to target inhibitors and or may differentiate over time, so maybe don't need such an aggressive approach. Okay, the third case is a 4-year-old girl. This girl had loss of vision and developmental milestones and had this huge enhancing supracellular optic pathway mass on imaging. So this was not possible to get out. So she just had a biopsy done. Morphologically looked very low grade, like a pyloid, very low proliferative index. So what do we know about low grade gliomas in the pediatric age group? So, as I mentioned earlier, this is the most common CNS neoplasm in children. They're histologically very diverse, and the classification of these is very complicated. It's a combined morphologic molecular approach, but a lot of these morphologies overlap, the molecular overlaps, and so they can be confusing. The outcome is variable. Mostly you're dealing with issues in terms of multiple progressions as opposed to death, but sometimes patients can die from this. And important to remember is they're distinct from the adult lower grade gliomas. So these are not IDH mutant, basically by definition. So it's sort of this side of the page of the classification of gliomas. As you can see, there's this introduction of these pediatric type diffuse low grade gliomas that was done to try to distinguish them from the adult lower grade gliomas. And there's a number of new entities that have been added here. There's also the circumscribed ones, which have been around for a long time and people are more familiar with. And then there's this mix of glioneuronal and neuronal tumors with a couple of new entities added here as well. So, it's important to think about, there's a lot of molecular here, but, what's more important, the molecular or histology? Do you always need to do molecular in these low-grade gliomas? And if you do the molecular, what does it mean? When we looked at our population-based cohort here at Sick Kids, this was about 1,000 low-grade glioma patients, which we collected over about a 20 or 30-year time period. About 2/3 of these cases are either arising because a patient has NF1 or because they have a BRAF fusion or a BRAF V600E mutation. And then after that, there's a bunch of sort of rarer type mutations that account for the rest. But each of these individually is fairly uncommon. And so that makes it actually hard to design a targeted approach to the molecular other than the big ones, the BRAF, the 2 big BRAF alterations. However, almost all of the alterations in these sort of converge on the RAS MAP kinase pathway, either because they have alterations up in one of these receptor tyrosine kinases, like the FGFR receptors or the PDGFR receptors, or they have alterations in PTPN11 or PI3 kinase, or down the RAS pathway. So there's evidence in almost all of these patients that they have activation of that pathway. However, if you look at, unlike some of the other entities where we see very tight associations between a specific mutation and a specific morphology, here it's more enrichments with a few exceptions. So if we look at pilocytic astrocytomas, the vast majority of them are going to have a BRAF fusion, but they don't all, and vice versa. It's not that every BRAF fused case looks like a pilocytic. So it is this bit of a mix and match approach, with the exception of a few entities that are really defined based on their fusions. So these are the ones with particular enrichments, and some are like, so obviously diffuse astrocytoma MIB or MIB-L1 altered is defined by having one of these alterations. Angiocentric gliomas should have a MIB fusion. The rest of them are more enrichments. They're not defining of those categories. So that's important to remember. And so I think the general approach for these should be using a layered diagnostic approach where you report both what is the morphology and what is the molecular, if you know what it is. So when do you really need molecular testing for low-grade gliomas? I think if you have like a morphologically classic entity, like a pilocytic astrocytoma in the posterior fossa that's been gross totally resected, you probably don't need to do molecular. Some of the situations where I've found the molecular characterization is helpful So you have a low-grade glioma in a young adult that's IDH wild type. Those, I think, deserve looking for a molecular alteration, because again, it'll change your approach. Is that a molecular GBM or is that a pediatric type glioma? If you have a case where you're considering radiating the patient and you might want further prognostic guidance, then it's really important. For example, we would very rarely radiate a BRAF fused patient in a child at Sick Kids. If you have a really small biopsy and you just aren't sure, is this a low-grade glioma or high-grade glioma, then the molecular can help. If it has a K27M, it's going to be bad. If it has a BRAF fusion, it's probably going to be good. Or if you have a growing, incompletely resected lesion with a potential for targeted therapeutics, that would be another situation where it's worthwhile finding out what is the molecular alteration. So this is our survival data. This is our low-grade glioma patients at Sick Kids. This is based on BRAF status. So this is progression-free. This is overall survival. NF1 patients are in green, BRAF fused patients in blue, BRAF V600Es in red, and then these are K27Ms in purple. These are all low-grade gliomas with K27. Obviously, they do poorly overall. But the BRAF fused and the NF1 patients have greater than 95%, and this is small, but this is a 20-year survival. So these patients survive long-term, and that's an important thing to remember. The BRAF V600Es are a little different. So you can see they do really well for about the first decade, and then it starts to drop off. So if you have a practice where you're seeing patients in their 20s or 30s, these can sometimes come back as high-grade gliomas once they've hit that 10, 15 years post their initial diagnosis. Other reasons why it's important to know. So even the response to standard chemotherapy can differ between different molecular subtypes of low-grade gliomas. So the BRAF fused and the NF1s are much more likely to respond to standard chemotherapy than the BRAF V600Es or the BRAF wild type cases. But the response to targeted therapeutics can be substantially better. So this is Jason Fangasero's study published back in the Lancet in 2019, where they used a MEK inhibitor on patients. This is at relapse. And showed that the response to a targeted therapy in patients that had already failed a standard chemotherapy was better than if you just gave them another standard chemotherapy. So again, this is a relapse patient. It may be important to know what's their molecular driver. For BRAF V600E, the data is maybe even better. So standard chemotherapy, these are waterfall plots. So if you're above the line here in green, it means you grew on therapy, and if you're below the line in orange, it means you had your tumor shrunk on therapy. So this is looking 6 months post therapy. If it's in orange, it means they had a greater than 25% objective response on imaging. So you can see this is the response to standard chemotherapy in patients with BRAF V600E. This is the response to a targeted therapeutic for patients with BRAF V600E. So if you have a patient with BRAF V600E who needs chemotherapy, which isn't everybody, they do much better on a targeted therapy if they can tolerate it. This is just one of these spaghetti plots showing that the response to the BRAF targeted inhibitors in these patients is generally quite rapid. So if they're going to respond, you're going to see that in the first 6 months. And then this is just some data showing response rate and what happens when you stop. So about 80% of patients will respond to the targeted inhibitor. Only 12% progressed on therapy. If you stop the therapy, almost 2/3 of patients will progress, but importantly, when they were re-challenged with the inhibitor, almost 90% of them responded when they were re-challenged. So important to remember for those patients. So just some generalizations about the others. There's a lot of details here and not really enough time to go into each of them. But in general, for pediatric low-grade gliomas, if it has a fusion, it is going to do better than if it has a point mutation. Fusions tend to be single drivers in children, as opposed to the point mutations, which you can see more than one. You tend to be younger if you get a fusion than the point mutations. And so overall, and you can see the overall survival here is in red for all of these patients do very well. The point mutation patients, it's a bit more variable. So obviously the K27Ms do poorly. The IDHs, it's kind of in the middle. You know, we originally thought, oh, maybe the IDH mutant gliomas that are arising in children means you found it earlier and they may have a longer survival than an IDH mutant glioma arising in an adult. That is not true. So your median survival is the same if you present at 15 as if you present at 25. The FGFR mutants are interesting. So these actually increase in frequency the older you get. So in the adolescent young adult population, this is accounting for almost 20% of the low-grade non-IDH low-grade gliomas, whereas the BRAF fusions become less frequent the older you get. These, if that's the only mutation you have, they can do quite well. But it's important to remember these are often associated with other co-mutations, and depending on how many mutations, some of these patients are ones that eventually succumb to their disease. So overall, the point mutations seem to be slightly different mechanistically than the fusions. So this is just to introduce a concept that for pediatric type low-grade gliomas, it's important to think about a lot of different factors when you're trying to judge, you know, what should your approach be. So the histology matters, the molecular matters, the age of the patient matters, and where is it located in the brain matters. And all of those are going to sort of combine into telling you what are the chances of survival for your patient. This is just sort of an easy approach to the testing. So BRAF V600E is an IHC. It's pretty easy. You can test your low-grade gliomas with that. If that's negative, you know, BRAF fusions are actually the most common alteration you're going to see in the pediatric age group. You can use FISH for those. So if you don't have access to NGS, that's an option. Obviously, in the midline, you would think about K27. If it's an older child or young adult, you would think about IDH. And then you're basically looking at NGS. So unfortunately for these, if you don't have one of the common ones, you're kind of stuck. If it's BRAF V600E, you should probably be looking for CDKN2A deletion similarly to the IDH, and that can again be done with FISH. So in this particular patient, we found a KI1549 BRAF fusion, and so she was treated with a MEK inhibitor and actually had a spectacular response. So, you know, we don't always get such a nice response, but if you do, you know, these patients, she did not have surgery. This was purely from the drug. So key things to remember. So about 60% of pediatric low-grade gliomas will have a BRAF alteration. About 40% of those are BRAF fusions and 20% are BRAF V600Es. The fusions are most common in the cerebellum and they're least frequent in the hemispheres. Most commonly look like a pilocytic astrocytoma, but you know, can look even like diffuse astrocytomas and still have effusions. If conventional chemo fails, these patients might respond to MEK inhibitors. For the V600Es, they're more common in the hemispheres. PXA, I think, is the most enriched for BRAF V600E, although obviously it's not the most common tumor. Gangliogliomas often have V600E, diffuse astros often have V600E, and then pilocytic sometimes, but they would be the least. These have a more aggressive clinical course and a poorer response to standard therapies, but may respond better to targeted inhibitors. So other alterations, each are individually rare. In general, the fusions do better than the mutations. FGFR1, TAC1 fusions are very similar in behavior to BRAF fusions. And then there's a whole host of other things, you know, that do well, like the FGFR2s, the FGFR1 tyrosine kinase domain duplications, the MIB and the MIBL1. These are usually epilepsy-associated hemispheric lesions, and they do well with surgery alone. They don't usually need additional therapy. Again, sort of the role for histology versus molecular is still unclear. Definitely the grading is still important. So if you think it looks like a high-grade glioma morphologically, then I would be very careful. And I think it's important that we're still reporting both our morphologic impressions as well as the molecular alterations. Okay, so that's sort of all I had. I want to thank all my lab who went and did a lot of the work that I showed, and thank you very much for your attention. Happy to take questions.
Thank you very much, Cynthia. That was great. There was a question or two in the chat. Find it here. So one question is that there have been some case reports of TERT promoter mutations in low-grade gliomas or pilocytic astrocytomas in particular that have then had an aggressive course. So could you comment on that?
So, you know, again, in pediatrics, that would be exceptional. Once you get older, then, you know, all bets are off, so to speak. So that's when you start getting into the, you know, what's called high-grade astrocytoma with pyloid features. Those can have TERT promoter mutations, more, you know, more commonly would have ATRX. And so, you know, again, I think, you know, in my experience, most of those actually morphologically look kind of more nasty than your typical pilocytic as well, although, you know, there are exceptions to that. So I would say once you know, get out of the standard thing, which is that BRAF occurs on its own, then it is possible that you can get things like TERT promoter mutations, allowing a pilocytic astrocytoma to behave in a more aggressive way. So, you know, our data would suggest that most pilocytic astrocytomas have a BRAF fusion. That's all they have. They eventually undergo oncogene-induced senescence because they do not have TERT or ATRX. And so they cannot, you know, maintain their telomeres. If they develop a way of maintaining their telomeres, then they become more problematic.
Great. I think that fully answers that question. I had put a question in about anecdotes of non-midline gliomas with H3K27 alterations doing not so badly, but it looks like you've answered that question with some real data.
Yeah, so again, once you're out of the sort of classic diffuse astrocytoma type of K27M mutation, then, you know, the survivals can be better. You know, like someone being cured, I have not seen. So, you know, 10 years, maybe 12 years survival with those circumscribed gliomas, non-midline ones, but you know, it's unlikely that you'll live forever with that.
Well, nobody lives forever, but okay. Any other questions? I haven't seen any others in the chat. Naren, anything you want to ask?
I'm okay, thank you. Thank you very much, Dr. Hawkins, for that comprehensive lecture. I've got a lot of messages being sent, whether you will be happy for your lecture to be shared on the YouTube for people to review later on. Would that be okay with you?
Sure. Yeah.
Oh, brilliant. Thank you very much. Much obliged. That's great.
Okay, so thank you very much, Dr. Hawkins. We're slightly ahead of time, but I think I can start with introductions and then the speakers can go ahead and have full time. Now, as I understand it, Naren, the next talk is recorded, but then the speakers will be online live for questions.
Yes, that's the plan.
Okay,
so I certainly have got their lecture. So
all right, well, let me give the introduction and then I'll let you go ahead and start the recording. So we're going to hear next about ependymomas. Dr. Michael Fehlings is an MD PhD who's professor of neurosurgery and vice chair of research at the Department of Surgery at the University of Toronto. He is the Jerry and Tutsi Halbert Chair in Neural Repair and Regeneration at that university. He earned his MD degree at the University of Toronto, did a surgical internship at Queen's University, and then entered neurosurgery residency at the University of Toronto. I believe he earned his PhD while a neurosurgery resident, which by itself is a remarkable achievement. He did some postdoctoral research after his training at NYU, first with Wise Young, who ran the research lab in neurosurgery when Joe Ransohoff was chair there. And then he followed that with a clinical spine neurosurgery fellowship with Dr. Paul Cooper. And these were in years when I was at NYU, so I interacted there and had the pleasure. I worked closely with Dr. Cooper and for many years and less closely, but episodically with Wise Young, and they were both great. He joined the neurosurgical staff at Toronto Western Hospital as his first faculty position and has gone on to the professorships and vice chair that I've mentioned. And he's going to share this talk with Dr. Nardin Samuel, who's an MD PhD, who, if the website is correct, is still a resident physician in neurosurgery at the University of Toronto, but has had an active research program from her graduate work involving, in general, I would say, molecular genetics of CNS tumors and hereditary tumor syndromes. So we're going to hear a recorded talk by these two esteemed individuals, and then they'll come on to the systems live to answer questions and comments. Naren, over to you.
Thank you very much, Dr. Mille. We have got Dr. Nardin Samuel. Welcome, Dr. Samuel. So I'm going to try to play your video. If there's any sort of, it will take me 2 seconds, and if not, I've got another backup system. It's all on hand. So let's see. Okay.
From the University of Hello, I'm Michael Fehlings from the University of Toronto Division of Neurosurgery, and it's a pleasure to be taking part in this symposium related to international pediatric brain tumor.
Can you all hear it? Dr. Mille, can you hear it? Great.
And Dr. Nardin Samuel from my team and I will be talking about the biology of ependymomas and emerging novel therapies. We have no relevant commercial disclosures, but we wish to thank our friend and colleague, Mr. Narenthiran, for this kind invitation. Much of what we'll be discussing today was recently published in this January 2022 article in Nature Reviews Cancer on the biology of ependymomas and emerging novel therapies. Right at the outset, I want to acknowledge the wonderful team of students and scientists that took part in this article, and which represent a part of the collaborative spirit inherent to the Division of Neurosurgery, to oncology at the University of Toronto. The first author on the Nature Reviews cancer paper was actually a medical student, Amer Saleh, and Amer is going to be going into medical oncology. He's just starting his internal medicine rotation. And that's his brother, Mohammed, who also collaborated on this. I'm indebted to the collaboration of Professor Michael Taylor, who I'm sure is extremely well known to you, world authority in molecular neuro-oncology. And we have two neurosurgical residents who are part of this paper. Dr. Kyle Jurashka is a student in Michael Taylor's laboratory, and Kyle is doing his PhD in molecular neuro-oncology. And I'm going to be handing over in just a moment to Dr. Nardin Samuel. And Nardin is a brilliant trainee in our neurosurgical residency program. She's an MD PhD graduate. She's currently in the middle of her training in the neurosurgical program at the University of Toronto, and she's doing an embedded postdoctoral fellowship as a surgeon scientist trainee in our program. And Nardin truly has a significant amount of expertise in molecular neuro-oncology. And so with that introduction, I'd like to hand over to Nardin to deliver this lecture.
Thank you so much, Dr. Fehlings. I'm just going to share my screen here. So thank you again, Dr. Fehlings, for that very kind introduction and for the opportunity to be involved in this project. And I also would like to acknowledge Dr. Naren for the opportunity to be part of this symposium on international brain tumors, and really among a very distinguished set of speakers. So we really appreciate this opportunity, and we hope that This presentation will give you an overview of the topic of ependymomas and really summarize our recent paper on this topic. So first, I'll just start with an illustrative case. So this is a case of an adolescent 16-year-old male who presented to his primary care physician with progressive weakness and back and leg pain bilateral for about 2 to 3 months. But acutely over the past 6 weeks, the patient was complaining that these symptoms were getting worse. And so he was sent for imaging, and an MRI spine, as demonstrated here, shows kind of an expansile mass in the lumbosacral area. That's an intradural intramedullary lesion. And so given this finding, the patient is referred for neurosurgical consultation and ultimately undergoes surgical resection. And so intraoperatively, there's extensive tumor that's found to be invading the dural walls and the nerves. And so as a result, really only partial resection was possible. And then the patient required postoperative radiation. On follow-up, long-term follow-up, so the pathology was determined to be anaplastic ependymoma, which is a grade 3 spinal ependymoma. And the patient was able to ambulate with minimal deficits, but there was extensive scar tissue as well that was noted, kind of caudal to the tumor. This is a second case study. So this is a case of a very young 12-month-old female that presents with a 7-day history of left-sided weakness. And again, kind of in an acute setting, brain CT demonstrates a very large frontal parietal lesion here. You can see this significant hypodensity on just plain CT imaging. That's measuring 7.6 centimeters in maximal diameter. So this patient also underwent surgical resection using a transparietal approach, and the pathology was determined to be grade 2 to 3 ependymoma, and the molecular work was pending. The patient then underwent intensive chemotherapy postoperatively for 16 months, and on follow-up, she was found to be neurologically intact without any recurrence. So these are just some illustrative cases here to show both spinal ependymomas as well as intracranial ependymomas, and also the type of epidemiology in the patients that we see these pathologies in. And I'll go through more detail about the heterogeneity of these tumors and really what we're learning about the molecular signaling pathways, and also what the opportunities are in terms of novel therapies in this field. So these tumors are quite rare, and it's estimated, at least in the pediatric population, that they would occur in a frequency of approximately 3 per 1 million. And the vast majority of ependymomas are intracranial, whereas comparatively, there's a much higher preponderance of spinal tumors, spinal ependymomas in adults with an average age of approximately 45 years at diagnosis. I think one of the most important things to note is that these tumors can be very highly recurrent. Over 70% of them do recur, and the recurrence is typically associated with a very poor prognosis and a short disease-free survival. And so pediatric patients actually are known to have much worse prognosis than adults for this particular pathology. So the goal of this talk is really to connect the various methods of investigation that have been done over the past century or more, and highlight what are the gaps in our knowledge pertaining to ependymomas to help kind of guide and drive future research in this area. So I'll go through some of the main figures here in our manuscript, and this is one of the first ones where we describe a timeline of the major discoveries that have been made in the field of ependymoma. So really, it was kind of this very early study in 1924, the Percival study, which was the most comprehensive study of ependymomas. And in the decades following that, there emerged the viral theory of ependymoma tumorigenesis. And around that time, this was kind of the dawn of the viral era of oncogenesis, kind of overall, among other tumor types as well. And then, as you can see here, it really wasn't until, almost half a century later that molecular type analysis was then subsequently being performed and then informing us about the biology of this particular tumor. And I'll go through some of these highlights in more detail in the subsequent slides. So the first topic to discuss is tumor heterogeneity. And this is something that I'm sure is very familiar to this audience, but is also a very salient concept, not just in ependymomas, but in oncology in general. But as it pertains to ependymomas, actually, One of the most interesting findings was really a landmark paper, landmark cancer cell paper in 2015 that identified 9 molecular subgroups of ependymomas based on DNA methylation profiling. And these ependymomas distributed by supratentorial, posterior fossa, and spinal tumors based on their location. And the ST or supratentorial SE subependymomas, and then the other supratentorial tumors were actually denoted by fusions, which I'll also explain in the subsequent slides. So fusions in YAP1 gene and the RELA fusion as well. Similarly, there's also stratification in the posterior fossa lesions. And this is actually very important because it had significant implications for prognostication. In particular, for example, the PFA ependymomas were known to have, were shown to have actually a much worse prognosis relative to the PFBs. And similarly, stratification of the SP or spinal ependymomas by molecular subgroups and DNA methylation profiling. When we're looking specifically at spinal cord ependymomas, one of the defining features, as we kind of describe in greater detail in the paper, is that there's loss of chromosome 22, 22q. And this leads to increased expression of genes, particularly in this area relative to what we see within intracranial ependymomas. And I believe this feature is actually present in over 90% of spinal ependymomas. So it's a highly common recurrent event. Similarly, across spinal ependymomas, myxopapillary tumors are unique. So we see them actually as unique in their location from a neurosurgical perspective, but also at the molecular level, they often demonstrate elevated expression of factors that are kind of driven by this so-called Warburg metabolic phenotype, or an alternative metabolism pathway that is different from the aerobic metabolism pathway. When we're looking at posterior fossa ependymomas, the two major subgroups are posterior fossa A and PFB, and this again has treatment implications. So I think this is a relatively important slide for focusing on posterior fossa ependymomas, because these are the most common ependymomas that we see in the younger age categories. And really the major summary of the work that's been done is this. So essentially the PFAs are primarily laterally located, and they're known to be aggressive and also highly recurrent. Again, at the molecular level, it's thought that there's activation of oncogenic phenotypes that lead to improved and enhanced cell migration and also angiogenesis. Again, kind of with this concept of heterogeneity, there's intratumoral heterogeneity among each group. So within even the subtypes, there are kind of further subtypes, which speaks to really speaks volumes to the importance of kind of personalized approaches to these tumors. So there are minor subgroups within the PFAs, and there are 9 of them, and then 5 minor subgroups in the PFBs. And then within the PFBs specifically, although they're overall kind of more histologically favorable subtype in comparison to the PFAs, loss of chromosome 13q is actually thought to be associated with a more poor prognosis in these patients. And then, of course, there's intratumoral heterogeneity, which is a topic that's really kind of been plaguing the molecular oncology community for the past couple of decades, particularly with the advent of sequencing technologies, where we're able to kind of use single cell approaches to really demonstrate that within one tumor are actually many different genetically distinct subtypes. And similarly, this is something that we see in ependymoma as well. There are structural chromosomal alterations, and these can actually occur before the accumulation of single nucleotide variants. I'll talk about this in a little bit more detail, but many of you may be familiar, for example, of the concept of chromothripsis or kind of chromosome shattering, which are very large scale chromosomal alterations that occur, and then subsequently point mutations occur. So over the next couple of slides, I'll go over the specific signaling pathways that have been shown to be relevant in the context of ependymomas, and really demonstrating that these are the potential pathways to look at in terms of future research, but also in terms of future development of novel therapies. So this is a table from our manuscript as well that demonstrates the oncogenes and tumor suppressors within ependymomas. And just looking at the intracranial lesions, you can actually see that the majority of the supratentorial deregulated genes are actually oncogenes, whereas in the posterior fossa ependymomas, we also see a significant deregulation of some tumor suppressor genes as well. And interestingly, there's not a lot of overlap between the particular genes and fusions that you see in the supratentorial compartment relative to the posterior fossa lesions. And I think this also speaks to, again, the heterogeneity of this disease, where, you know, these are all ependymomas, so to speak, but actually their location is quite dictated by their genetic makeup as well. So one of the highlights, for example, that we see in the supratentorial ependymomas are the ZFTA ReLA fusion proteins. And this is thought to be very important in the NF-kappa beta signaling pathway. As I mentioned earlier, the concept of chromothripsis. So the theory here is that chromothripsis, or large-scale catastrophic chromosomal shattering, occurs, and this is kind of an early event. And then this results subsequently in the fusion between these two genes. And this fusion here ultimately activates expression of genes that are important in modulating the NF-kappa B signaling pathway, which is an oncogenic signaling pathway. Similarly, this is another implicated pathway, which is the YAP1-MEM-LD1 fusion. And this, so the Hippo pathway, it's in and of itself is actually tumor suppressive. So it's thought to limit tumorigenesis. But this fusion then subsequently disrupts Hippo signaling, and then again, kind of decreases the signal that's associated with hampering tumorigenesis in this area. And then again, this can promote tumorigenesis. And then finally, most recently, one of the areas that has come up is MYC signaling. So, you know, many of us know that MYC is a very common oncogene that's commonly mutated across many human cancers, but it's really only recently that it's been implicated in the supratentorial EPN subtype. And MYC is a well-known and very, very well researched transcription factor that promotes cell proliferation, differentiation, among many of its other posited activities as well. And specifically, MYC-N is thought to be amplified in a particular aggressive subtype of the spinal ependymomas, particularly occurring in adults. And then also, of course, there's EGFR signaling, which again, many of us are familiar with. And so at least in this context, what's thought to occur is that there's increased co-expression of EGFR, so ERBB2 and ERBB4. And this co-expression then leads to proliferation and diminishes, overall is thought to diminish prognosis in pediatric ependymomas, which is specifically the presence of increased EGFR expression. There are also various oncogenes that promote continuous EGFR response. So essentially, this is kind of where the EGFR pathway is consistently in the on state. And then again, this has significant implications in the clinical realm because, of course, there are targeted therapies against EGFR. And the PIK3 kinase AKT pathway also functions quite similarly as well, as shown here. And elevated levels of phosphorylated AKT activates PIK3 kinase signaling. And then again, this leads to greater cell proliferation. Similarly to EGFR is associated with worse overall prognosis. Similarly with Notch. So essentially, I'm just highlighting some of the key aspects of this figure here. Notch signaling as well. As you can see here, it's particularly associated with pediatric ependymomas. This is not something that we see as commonly in the adult setting. So those are the oncogenic pathways that have been identified through the various research papers that have been published over the past couple of decades. And now kind of looking at tumor suppressor pathways. One kind of not so surprising is the P53 pathway, which is essentially implicated in every human tumor type, whether it's adults or in the pediatric population. And P53 is notorious as a tumor suppressor. Specifically, it arrests cell growth and induces apoptosis. And overexpression of this particular protein here, MDM2, has been thought to occur in the context of ependymomas. Similarly, cell cycle regulation. So these are very canonical pathways that have been described also again for many cancers outside of the context of CNS tumors. And the pathways by which these are deregulated are very much the same. So for example, silencing or deletion of CDKN, which is a tumor suppressor gene, leads to further cell cycle dysregulation. And in the context of ependymomas, this has been associated with a worse overall prognosis. NF2 signaling as well has been implicated, particularly insertion or deletions or mutations within the NF2 gene have been identified in up to 40% of spinal ependymomas. And then turning to epigenetics, which I believe we'll hear a lot more about DNA methylation from one of the other panelists, Dr. Ken Aldape, who's an expert in this area. But epigenetics is really has been a very hot area of research within the CNS setting, because essentially what can't be explained by genetic analysis and tumor sequencing, significant insights have been garnered through the study of epigenetics in this area. So in particular, when we're looking at epigenetics, this speaks to aberrant methylation for overall. There are other epigenetic pathways, but kind of on a very simplified scale, looking at methylation. And so when we talk about essentially what it means is that there are certain areas of the genome which are known as CPG islands, or they're kind of continuous regions of CG dinucleotides, typically occurring in the promoter region of genes. And when these are methylated, this leads to inactivation of the gene and essentially decreased expression. And this is something that's been seen to be crucial in the pathogenesis of ependymomas. Up to 20% of the higher grade ependymomas have a methylated tumor suppressor gene. Similarly, promoters are methylated to inactivate TET proteins as well. Also looking at non-coding RNAs. So we've been talking a lot about so far the different signaling pathways and proteins that are affected through fusions and mutations and so forth. But kind of on the non-coding level, at the regulatory level of the genome, there's also microRNAs and long non-coding RNAs, among other non-coding RNAs that are implicated. Again, these are kind of broad oncology concepts, but things that we also have seen in the context of ependymomas. So differential expression of microRNAs within ependymomas, and also long non-coding RNAs, which modulate expression of various genes, either locally, kind of in cis or in trans, on a more broad scale as well. And then finally, one of the things that we highlight is multi-step tumorigenesis. So of course, there's kind of the classic teaching where there's kind of this two-hit hypothesis, and over time, the genome kind of accumulates mutations, and this leads to the progression or continuum of tumorigenesis. And really, in the context of invasion and metastases, one of the interesting things about ependymomas is they're, as I mentioned, they're very highly recurrent, and they recur locally. They may also spread through the CSF. This is less common, but also possible to occur. And it's very rare, similar to other CNS tumors, for them to metastasize outside of the CNS. Specifically, elevated expression of certain genes known as matrix metalloproteinases, so MMP2 and MMP9, correlate with a shorter survival in the pediatric population. And this is just a snippet here from one of the tables in our paper. So we talked a lot about nitty-gritty details of the different genes that are dysregulated, and the different mechanisms by which they're dysregulated, also kind of doing a very quick overview of epigenetics, and also kind of coming back to the beginning of the talk, discussing the importance of tumor heterogeneity and how can we tie all of these things together to kind of help drive research forward in this area. And also kind of translate these findings into actual molecular therapeutics for patients. So kind of the mainstay of treatment for ependymomas, both intracranial and spinal in pediatrics and in adults, is surgery and radiation. So standard treatment is surgical resection and gross total resection when possible. And typically there are some kind of subpopulations where patients may not undergo postoperative radiation. But for example, in the case of subtotal resection or in certain molecular subgroups, postoperative radiation is currently the standard of care. And about half of pediatric patients, I think that it's about 50 to 75% of pediatric patients do have a local recurrence, even in the presence of complete gross total resection. And it's really kind of following these relapses that further surgery can occur. And at least also in the neurosurgical realm, sometimes there's a concept of a second look surgery. So essentially the first surgery is conducted, and if a gross total resection is not able to be achieved, then subsequent surgery is then performed as well. But again, in the context of relapses, this is associated with a much worse overall survival. And following the relapse, surgery and radiation actually can delay the disease progression, but it doesn't really impact the overall survival of these patients, which is quite a devastating outcome. And so really in terms of chemotherapy, so there are chemotherapy regimens that have been studied, and there's dose-dependent study of temozolomide in particular, which, as we know, is used in the adult GBM population. It has shown some promise in recurrent lower grade as well as the grade 3 supratentorial ependymomas, some infratentorial ependymomas and spinal cord ependymomas as well. These are active ongoing clinical trials. And it's been proposed in terms of salvage therapy for patients with recurrent ependymomas. Our paper summarizes the ongoing clinical trials in this area as well. And so there are, of course, challenges. And what are the challenges for us to translate these therapies? Of course, one of the main considerations is effective drug delivery due to the blood-brain barrier. But this is not a new problem. This is something that we've been facing for many CNS lesions for many years. And so there are ways, novel ways that we're working on overcoming the blood-brain barrier, both with direct and indirect approaches, for example, focused ultrasound and so forth. In addition, as we mentioned, tumor heterogeneity is actually quite significant. It presents a challenge to actually tailoring treatments to these patients. And I think that's, in my view, that's actually one of the most difficult challenges, because it's almost like you're kind of slicing a pie in so many ways that you can't slice it further. It's almost like every tumor is very unique and distinct. And I think although there's a lot of potential for personalized medicine, that does present a challenge in terms of developing novel therapies. And also there are a lot of preclinical models that have been conducted in, for example, animal models and so forth, but they often don't accurately predict clinical response. So, overall, these are not new challenges, and these challenges are not unique to ependymomas, but given the fact that ependymomas are such diverse tumors, these challenges actually become even more amplified in this setting. And so in terms of opportunities, firstly, tyrosine kinase inhibitors, there are a number of preclinical studies that have suggested that they may have therapeutic potential in this domain. And they've demonstrated effectiveness, for example, in ependymoma cell lines in terms of decreasing cell viability and increasing apoptosis as well. Telomerase inhibitors. So there have been preclinical models looking at specific telomerase inhibitors and demonstrating that they can decrease the proliferative index of, at least in cell lines. And these studies are primarily on established ependymoma cell lines. We talked a little bit about angiogenesis and EGFR. Also, bevacizumab is an accepted and FDA approved agent for various other tumor types that functions via the anti-VEGF pathway. And it's also been used in various cancers. There are some phase 2 clinical trials that were ongoing in the ependymoma sphere, but unfortunately they have not demonstrated significant effects. And finally, there's immunotherapy, which has been challenging due to, again, penetrating the blood-brain barrier and significant toxicity that's associated with various immunotherapy approaches. There have been the dawn of kind of immune checkpoint inhibitors as well have been shown to potentially have therapeutic benefit. And there are many strategies. So the immunotherapy approach is a really multi-pronged approach where there are specific agents to induce checkpoint blockade, but then there are also novel therapies such as CAR T therapies and oncolytic viruses and so forth. And these are all currently in active clinical trials. And they have shown some efficacy in other tumor types as well, which is somewhat promising. So overall, I hope that we've been able to demonstrate that although this is a very rare tumor type, it's quite diverse. It seems like we know quite a lot about it. And in us synthesizing this paper, we learned that there's a whole host of molecular research in this field, but really what the gap is and what's missing is translating this and figuring out what's the best way to actually harness this knowledge to improve the prognosis of patients. And I think one of the things I want to highlight as well is that these molecular studies have really helped in informing prognostication of these patients. And so really one of the next steps is going to be continuing to push the envelope to implement novel therapies that are based on the molecular studies that have been done. So with that, I'll end there. I'd like to thank you very much for your attention and again for this opportunity. And again, like to thank Dr. Fehlings and Dr. Nardin, and we'd be happy to take any questions.
Dr. Mille, Dr. Mille, it's to you. Dr. Mille, you should be able to unmute your microphone now.
Now I can. I couldn't.
Yeah.
No,
Sorry,
yes.
sorry about that.
Thank you. Dr. Samuel, thank you very much for that excellent talk, and Dr. Fehlings for the introduction to it and for facilitating it. That's really great, and for the audience, we can see how this follows on from the classification talk that I gave early this morning, greatly amplifying the individual areas that I went over very briefly for ependymomas. I didn't see any questions in the chat, but perhaps I missed one. Just some thanks.
Can I ask I think a more general question, but hopefully a specific question after that to both Dr. Samuel and Dr. Fehlings. I'm keen, very keen that neurosurgeons become very familiar with molecular biology, because I have seen what happened to aneurysm surgery, and certainly in Europe, in Britain, that the endovascular interventionalists are now doing it, and we have kind of become on the side. And I remember I had a chat with Professor Andre Grottenhuis about 5 years ago, and he said to me, Naren, if neurosurgeons don't become familiar with the molecular biology, then we will end up being people who are technicians, who are just putting a burr hole and giving medications. And how do you, first of all, I can come to Dr. Samuel, how do you see yourself learning from this, your experience of molecular biology and neurosurgery, to take it further when you are in practice, and then a global idea from Dr. Fehlings? Thank you.
Thank you, Dr. Nardin. Thank you for that question. And I completely agree. I think that there's a lot to be said about, not being viewed as technicians, but really having a solid understanding of the pathologies that we're treating, from a surgical perspective as well. So, I'm very grateful that I had some dedicated research time before residency and now during residency as well. And I think that really immersing yourself in research is something that's very beneficial, particularly in the area that you want to ultimately subspecialize in. So that would be kind of my main perspective, but I completely agree about that sentiment. And sometimes it's not easy to keep up with the literature in an area that you don't know much about. And I see that, residency programs are moving towards more dedicated research time, whether it's, for a PhD or for smaller research blocks. And I think that should also be emphasized in the curriculum as well for the next generation of neurosurgeons.
Thank you.
So Naren, thank you for the question, and thank you for the kind invitation to participate. And I fully agree with you that neurosurgeons need to be aware of the advances that are occurring in molecular biology, and this has particular relevance neuro-oncology, but also I would argue many other areas in neuroscience and in neurosurgery. I would say that there are opportunities for surgeons who are in practice to embrace the expanding knowledge that's occurring. That was in part our desire to come up with this article, because ependymomas are in a sense a little bit of an orphan disease. They're very important, they're very debilitating, but remarkably understudied, particularly for spinal cord ependymomas, which is the area that I focus on. So I fully agree with you. And then I think that the model that we've tried to aspire to at the University of Toronto is this concept of formal training and research. So we have this surgeon scientist training program. And I wanted to highlight, Nardin Samuel, because she did her PhD in molecular oncology. And I think this would be a wonderful opportunity to also highlight a star example of our training program.
Thank you, Dr.
If I
Fehlings.
can comment, Naren.
Sure, please.
The model of care that I think we're all trying to espouse, at least where I've been and where I've talked to other people, is multidisciplinary care. These are complex diseases that no one person is just going to handle by himself or herself, whether they're a neuro-oncologist or medical oncologist or a radiation therapist. And so it's very helpful when you have a meeting of all these different specialties that everybody knows something about the molecular pathology and the molecular oncology of these tumors, so that it's not just one person offering their recollection or their opinion. To have everybody have a discussion and say, that's exactly what this tumor is, and that's what we expect of its prognosis based on available data.
I think you've said it beautifully. Couldn't agree more. And for myself, it's I really enjoy interacting with the colleagues in the various multidisciplinary conferences. Sometimes we're fortunate, the patient's fortunate, we're able to eradicate the disease with surgical approaches. But as Nardin showed, with the two illustrative cases, surgery has a role, but there's other forms of treatment that are critically required.
Thank you very much. And I hope you are okay for the recording to be available, because these are complex areas, and I'm sure myself as well as many of the delegates will love to go through the lectures. And then if you have any questions, to send you an email, if that's okay, Dr. Simon.
Absolutely. Would be absolutely fine. Just send us the link and we'll forward this also through our networks, if that's okay.
Oh, that's brilliant. Thank you.
Very good. Okay, thank you again for the invitation, and thanks to you, Nardin, as well.
Thank you both. Thank you, Dr. Fehlings. Thank you, Dr. Naren.
Thank you very much, Dr. Samuel. Thank you, Dr. Fehlings. Dr. Mille, it's to you. Thanks.
So, Naren, we're a bit ahead of schedule.
Oh, that's good. That's good.
Do you want to go on to your talk right now, or do you want to take a break?
What do you call? I'm happy to go ahead with my talk. Absolutely.
Very good.
Good.
Okay.
So let me just first of all try to share it.
Well, let me introduce you briefly.
Thank you. Thank you.
If there's anybody on this call who does not need introduction, it's Naren, who is I'm not going to go through his academic background because we'll be here all morning or all day. He's just done a plethora of things, and I'm just amazed at how he has virtually single-handedly built this Listserv endeavor into this manifold thing where there's a research Listserv, there are courses, there are master classes, there are research endeavors underway now, collaborative research efforts across continents. And it's just amazing that he's done all this and is still a practicing neurosurgeon on the side. So we all know who he is and what he's accomplished, and I think it's extraordinary. And I'm grateful that he's willing to step in and talk to us about medulloblastomas to round out this course for day one. Over to you.
Thank you very much, Dr. Mille, for that very kind introduction. It has been an absolute pleasure knowing you for the last 10 or 15 years and learned a lot. And in terms of this talk on medulloblastoma, I just want to, this is not going to be a talk as that has been given previously by experts in the field. This is from a neurosurgeon's point of view. And so now I will try to share this presentation. Just a second. Good. Let's see. Can you see that, Dr. Mille? Can you see the slide? Great, thank you. So I'm just going to take you through some basics of the molecular biology of medulloblastoma. This is to make sure that this symposium is covering all the main topics. So as Dr. Mille mentioned earlier, with the histological types, there were the three main types with the classic desmoplastic nodular, and the subgroup of that being the medulloblastoma with extensive nodularity, and then large cell anaplastic medulloblastoma. So these were the histological classifications, and they were not so great in terms of predicting outcomes and tumor response. So in the molecular analysis that was published by Taylor and team, Bill Taylor from Toronto, certainly ushered in this molecular biology in pediatric tumors. And in the current WHO classification, they have divided the molecular subgroups of medulloblastoma into wingless activated, the sonic hedgehog activated with wild type P53, and sonic hedgehog with P53 mutation, and the non-wingless and non-sonic hedgehog, which encompasses the group 3 and the group 4. So these are the main subgroups with the molecular subgroups for medulloblastoma. So if you just look through the wingless activated medulloblastoma, then it's the least common. And in terms of the histology, it corresponds more to the classic histological appearance, and metastasis is rare. And in terms of the age of the patients who have wingless medulloblastoma, it's absent in infants, but mainly in older children, about median age of about 10 to 12 years. And in terms of the sex distribution, same between males and females. And while it has a very good prognosis for the under 16-year-olds, with excellent prognosis of 90% 5 years survival. And in terms of wingless subtypes, then you have got the wingless alpha and wingless beta. And the main thing with the wingless alpha is that in chromosome 6, there's monosomy, whereas in wingless beta, it's more diploid chromosome 6. And the wingless beta is more in adults. However, in terms of the survival, they both have similar survival, but in adults, when these subgroups are found, then the prognosis is poorer than in children. In terms of wingless activated medulloblastoma, the common genetic alterations are the mutation in CTNNB1 gene. It's in about 85 to 90% of the wingless activated subgroup, and also the chromosome 6 monosomy in 70 to 80%. However, at least about 5% of the wingless medulloblastomas don't have these two abnormalities. So we can't just rely on these two for it to say it's wingless, because you can have aberrant chromatin regulations in this subgroup, And although there is a frequent TP53 mutation in this subgroup, they don't have much prognostic significance. Wingless medulloblastomas can be inherited from germline mutations. Certainly in Turcot syndrome, there's a germline mutation of the APC gene on chromosome 5. And the other germline mutation that can lead to a wingless type medulloblastoma is anaplastic lymphoma kinase gene. Now, if we move to the sonic hedgehog activated medulloblastomas, it accounts for about 30% of the medulloblastomas. The prognosis is more intermediate with a 5-year overall survival of 70%. And here, the patients who get this are more infants and adults and fewer in the childhood cases. The histology for these patients with the sonic hedgehog can be cover pretty much all the variations of the histological types. And in children with sonic hedgehog activated, then you do see the more of the large cell anaplastic type. And with the if it was sonic hedgehog activated and it's desmoplastic histology type, then there's increased survival in infants. If you look at the sonic hedgehog types and look at the germline mutation, then we can patients with live Fraumeni and Golin patients can develop medulloblastomas with the sonic hedgehog type. And in live Fraumeni, there's hereditary P53 mutation, and this accounts to about 20% of the sonic hedgehog type medulloblastomas. The commonest age is around 5 to 16 years. And because in these patients there's high rate of mutation, radiotherapy can actually accelerate growth of the tumor. In Golin syndrome, the mutations are on PTCH1 and SUFU genes. And in these patients, radiation therapy can increase secondary tumors, including skin cancers. In terms of the gene mutations, as I mentioned, the PTCH1 is about 43%, similar in all age groups. However, with the SUFU mutation and SMO mutation, the SUFU mutation is particularly in infants and adults, whereas the SMO is in about 90%, particularly in infants and adults as well. So in SUFU and SMO, they are more in infants and adults, but PTCH, that you get a distribution over the age group. If you look at the sonic hedgehog medulloblastoma and the TP53 mutation, the gene that produces the P53 protein, then as Dr. Mille said in his talk, if there is wild type P53, then the prognosis is favorable, 81% of 5-year overall survival. However, if there is a mutation in the TP53, then the prognosis is poor, with about 40% 5-year overall survival. The other mutations, so there are many other mutations, but the other main mutations are in the TERT promoter and mutations in the pathway of PI3K, AKT, mTOR pathway. So now if you go to the last group, the group 3 and group 4, then they account for about 25% of the cases. The peak diagnosis is around the young child, 3 to 5 years. And the worst prognosis, they carry the worst prognosis. In infants, the 5-year overall survival is about 45%. And in children, the 5-year overall survival is 58%. So they are pretty poor outcomes. Here, the males are more prone to have it. And once again, in terms of the genetics, it's a heterogeneous group, but here germline mutations are rare. And one of the genetic event that's seen early in these tumors is a tetraploidy. If you just look at the group 3 medulloblastomas, 40% have the large cell anaplastic type, and there's MYC amplification. And if there's a MYC amplification, then the prognosis however, whether the patients with group 3 have metastasis or not does not necessarily affect their prognosis, because the prognosis is quite poor in this group. If you look at the group 4, it's most prevalent, 45% of the childhood medulloblastomas and 25% of the adult cases. Here the prognosis is intermediate between 75 to 90% 5-year overall survival. Here again, the males are more likely to have it. And one of the favorable prognosis with group 4 is if there's a chromosome 11 loss. In such chromosome 11 loss, there's a 5-year overall survival of over 90%. If the patient has, these are very associated with metastasis. 30 to 40% of the cases at presentation have metastasis, and in those cases, the prognosis is poor, around 60% over 5 years. So the talk that I have given you is mainly from this paper that was published in 2020 by this group from Hungary. I will definitely recommend you to have a look at it. And they have a quite nice summary diagrams, which I'm reproducing here, which gives you the distribution of the subgroups of medulloblastomas and what are the common genetic abnormality and the overall 5-year outcomes, 5-year survival. I think this is a good reference to have. And what they have gone in their paper is to propose a risk stratification system. I'll just take you through how they have gone about it. So if they on the row on the first column, you have got the 4 different subgroups: the wingless, the sonic hedgehog, group 3 and group 4. I'm purposefully repeating it because for neurosurgeons it's nice to hear these groups again and again for us to get familiarized with. Then here they have divided into 4 risk stratification. Those patients with low risk, and then it goes across to high risk with poor outcome, and then the groups of that tumor where the outcome is that they are not so sure about. So if you look at the wingless ones, then they are usually metastatic and usually under the age of 16, and it has low risk and survival is high. And if you look at the sonic hedgehog type, they have divided that into 3 risk stratifications. The non-metastatic TP53 wild type and no MYC amplification under the standard, and the MYC amplification non-infant metastatic still TP53 wild type as high, but once you have TP53 mutation, then they stratify it under high risk. And this goes on for the group 3 if it's non-metastatic and no MYC amplification standard, but if it's metastatic and MYC amplification, then they stratified as high risk. And for group 4, once again, if it is non-metastatic with that chromosome 11 loss and chromosome 17 gain, then that has a good outcome. And so they have stratified under low risk. But if it was metastatic and no chromosome 11 loss standard, and if it is metastatic, high risk. So once again, I think whether this proposal will be accepted by all the people in neuro-oncology neurosurgeon will be away to see, but it's certainly a good reference to have. And this is from the paper by Louis et al. On neuro-oncology from last year, where they published the summary of the new WHO classification for CNS tumors. For medulloblastomas, they give the three subgroups: the wingless activated, the sonic hedgehog, and the medulloblastoma with non-wingless and non-sonic hedgehog. And then they have given the various gene mutations and molecular profiles that are associated with this. So once again, I think that will be useful for neurosurgeons to have before going to MDTs. So in conclusion, I think for a neurosurgeon, a modern neurosurgeon, a neurosurgeon of the 21st century, I think understanding molecular biology is, I find it gratifying, and it's not difficult if you start from the basics. And I'm planning to have small bite-sized tutorials on people to get, start from DNA and then work up. So I hope that you guys who have knowledge in this can help me. And the knowledge of the molecular classification then leads to the risk stratification. And with the risk stratification, then you can decide for the low risk tumors, then you can try to avoid treatments that have more complications or more risk of cognitive damage, like radiotherapy. So these things have to be clarified. And once again, at the end of the day, the aim is to give a personalized care for the patient with the least amount of adverse effect from the treatment. So thank you very much for listening to this talk. I hope this was useful. I appreciate I'm not an expert in this area, but I hope I wanted to cover this gap. Thank you.
Thank you very much, Naren.
Thanks, Lord.
So let me offer just a couple of observations on this and then sort of wrap up the course. For medulloblastomas, these are classified by the WHO as grade 4 tumors, just as glioblastomas are called grade 4 tumors, and yet the outcomes are very different. And so one thing to understand is that a grade number doesn't mean the same thing unless it's in the proper context. For medulloblastomas, if you think of the pre-molecular era, which wasn't very long ago, as several people have commented today, Michael Noah Hart, a neuropathologist of considerable esteem and a longtime editor of the Journal of Neuropathology and Experimental Neurology, published a paper, I believe in the mid-1980s or maybe early 1990s, in which he just did a retrospective review, I think from SEER data, of all the survival of all children, on all adults with medulloblastoma. And overall, because of the various incidences that Naren has so nicely shown us, the overall survival was about 80% at 5 years, 75 to 80%. And even at 10 years, it's not that bad. So this is not the same kind of dismal prognosis, even though we should still strive to do better. As Naren mentioned, the risk stratification is now very important. And while it may not have all the fine details of what he just showed, and there are some other factors involved, such as extent of resection, certainly the cooperative groups are now looking at reduced treatment for those who have the lowest risk strata for medulloblastoma. So for the Children's Oncology Group, I mentioned this earlier this morning, we now have an ongoing protocol that's been accruing patients for about 2 years now for children who have wingless or Wnt-driven medulloblastomas. They have to have no metastatic disease. They have to have had a near gross total resection, and there's an actual measurement for that. So instead of a percentage of tumor left, it's actually a volumetric analysis of how much tumor is still visible by MRI. The MR protocols are now very strict to really rule out metastatic disease. It's not whatever the institution wants to do, but there are prescribed protocols for how they do the MRs, and there's central review of that, just as there's central review of the pathology. And then there's the molecular studies that go on after that that point to the particular mutations. identify that the child is having a Wnt-driven tumor, and then the child gets reduced radiation therapy and reduced chemotherapy. There's a protocol in preparation for COG, which has been working through the process for about 2 years and should probably open within the next 6 to 12 months for non-Wnt low risk, so mostly hedgehog-related tumors. And again, we're excluding the anaplastics, but we're accepting the classics and for that one, we'll accept the nodular desmoplastics. So risk stratification is indeed not just the future, it's now. This is what is happening. And I think that's very, very important. So if I can, I'll wrap up what we've done since 7 a.m. my time. It's now noon. So we've been working 5 hours.
Thank you.
We started with an overview of the classification from me to give you a foundation. We then talked a little bit about methods and how they apply in clinical neuro-oncology. So how the molecular methods are applied to treating patients in the real world. We heard from Dr. Aldape about the methylation classifier and its power to help us to further refine diagnosis and resolve some difficult diagnostic questions. And then we went through particular types of tumors. So Dr. Hawkins talking about low and high-grade pediatric gliomas, and our nice talk about ependymomas from Dr. Samuel, and Naren's talk now on medulloblastoma. And I think we've hit all the major points then. Obviously, we could go on for days with all the subtypes and all that, but that's not a feasible endeavor. I thank you all for coming. It's been a privilege for me to be asked to help organize this by Naren, and I'm really impressed that he's thrown this together this way. Over to you.
Thank you very much, Dr. Mille. Thank you very much for putting together this fantastic day one on molecular, on neuropathology and molecular biology. I think this is the foundation, on which to practice neuro-oncology as well as neurosurgery. And sometimes we don't, as neurosurgeons, have this opportunity to get this teaching from as experts as you. So I appreciate that in your very busy practice, running a department, and in the short notice, you kindly, for your patience and putting together this fantastic meeting. I think I'm sure the neurosurgeons and neurologists here will greatly appreciate it, and this will be reviewed several times. So thank you very much, Dr. Mille, and I thank all the speakers for taking their time once again at a short notice to have this masterful lectures to educate us. Before I thank the participants, tomorrow we have lectures on all the major pediatric tumors from experts from around the world, from Japan all the way to Brazil, covering Europe and North America along. It should be fantastic. And then the day 3, we have got keynotes lectures from legends of neuro-oncology and neurosurgery. And on day 4, on Sunday, we have a controversies section. I hope this is the first of these international pediatric brain tumor course, and I hope that we will do it yearly. It might be others doing it, but I'm happy to facilitate it, and so that we keep up to date. And I look forward to your help, to Dr. Mille, as well as everyone in the participants to do that. Thank you very much to all the participants. It is for you that this is done, and it's a privilege and honor to host you. And I hope that you will all join us tomorrow and for the subsequent days. I wish you a nice rest of the day wherever you are. Bye for now. Thank you very much.